Rumisa madam, welcome.
Thank you, Dr. Karthik.
How are you?
Fine.
How are you?
A warm welcome on. Program will start within few minutes. Please wait. Dear participants, please wait for few minutes. A warm good morning, one and all join with us. I welcome all of you to the second day of national level online conference on innovations, pharmaceutical and biological sciences in association with Kalinga Biomedical Research Institute and Training Establishment, Odisha. Now we have Dr. Karna Reddy, Senior Associate from Pharma Industry, to deliver this topic, Scope of Regulatory Affairs. I would like to request Dr. Karthikeyan, Editor, Sydus Publishing House, India, to introduce today's speaker. So please.
Good morning to all and one and all. So I hope that you enjoyed the day one, and there is two brainstorming sessions of the novelties and innovations in that of your pharmaceutical sciences we have seen yesterday. And today we are going to see about that of your scope of regulatory affairs. So today I'm just going to introduce today's speaker, Dr. Karna Reddy. He has completed his PhD in JNTU Anantapur in 2019, MBA from SV University, India in 2011, M from Pharmaceutics from SRM College of Pharmacy, SRM University in 2007, B Pharm from Bharati College of Pharmacy under Rajiv Gandhi University of Health Sciences, Karnataka. Currently, sir is working as a senior associate in pharma industry from 2015 to till date, and he has worked as an assistant manager, regulatory affairs and quality assurance, biochemical and synthetic product limited, Hyderabad, India, from 2012 to 2015, and he works as a senior executive regulatory affairs from 2011 to 2012, and worked as an officer at Hetero Drugs Limited as in the regulatory department from 2009 to 2010, and he also worked in the Hetero Drugs US FDA and WHO approved plan, engaged in manufacturing and solid and liquid dosage forms, handling of dosages of US FDA approvals in Canada, healthcare approvals. He has presented 7 papers in national conference. He has professional experience in applying and approving applications for a drug license, testing license, loan license, and other lesion works, and also drug control department. Handling of technical queries and technological transfer to that of a product launching, compilation and submission of NDA 505B and ANDA products, CTD and eCTD format for that of US FDA for the solid and liquid dosage forms. Field of CBSE 30 years, approval for AFDA and a preparation of response amendments in timely manner for NDA, as well as he filed prior and post approval supplements and annual report for NDA products as per the guidelines. Reporting of ADR and launching notice, review of technical documents like PDR and dissolution profile, BMRs, BPRs and stability data, update labels and patents and exclusivity statement, and coordinating and compiling the response of FDA deficiency, compilation and submission of ANDs, products as CTD format, and Health Canada response to queries timely manner for submit of AND products. Coordinating consent department for procedures and document compilation of disorders and meet the submission timelines. With this brief introduction, I would like to invite Dr. Karna Reddy to deliver the talk on scope of regulatory affairs. Sir, please.
Yeah, thank you very much, Dr. Ken and Deepa, for inviting me for this session. So now, good morning to all. So now I'd like to share my screen and I'll explain further. So what is the scope of regulatory and what is the importance of the regulatory affairs? So how about the careers in regulatory affairs, and what are the things to know, and how we should prepare before getting into the industry? So just I'll give you a brief introduction initially, just what are the opportunities in industry first. Then I'll go on explaining regulatory affairs. See, there are so many opportunities in field of pharma industry.
Sir, please make it in the slideshow mode, sir, actually.
Slideshow, okay, yeah, fine. So now is it clear, Karthik?
Yes, sir, you can proceed. You can proceed.
See, there are so many opportunities if you have sound knowledge and commitment and sincere to the work. So you can grow in any of the field or any of the interest you have based on your knowledge and the experiences. So you have so many departments in the before establishing the regulatory field as today. Just when we came out from the graduation, so there is no much more establishment on regulatory affairs. So when we came out, we have these fields, this formulation development within the plant area, formulation developments and analytical. So regulatory affairs, there are only few. Those who are in exporting facilities, they are working on, or those who have an idea on how to get the registrations from the foreign countries, they are having the internal departments as the regulatory affairs, and technology transfer and packaging and development, manufacturing, quality assurance, clinical trials, and BAV study. And IPR also came parallelly with the regulatory affairs only. Then strategic sourcing. So without API, no one can manufacture any formulations. Then the business development. Once the regulatory and IPR came into the picture, then there was a chance for business development also. Then after that, once the regulatory field was establishing, by the time when so many CROs came into the picture, and they have divided and they have grabbed the opportunities in different fields like clinical management as a study coordinators and medical devices, medical imaging, medical scientific writing, regulatory operations. So we'll explain further on regulatory operations and all. So then in recent nowadays, in the past from the 5 to 10 years, project management is also one of the great opportunities, those who are having the knowledge on the basic core knowledge. See, basically drug discovery research, which is highly dependent on so without these biostatisticians and biochemical and molecular biological, so whatever the fields required for the chemistry or analytical purpose or the pharmacokinetics or the pharmacological activity, so all together which all the departments involvement is required to output of any of the formulation or the API. Just before the coming to the formulation, they'll go for the API testing for the thing. So where you need the toxicological studies, pharmacokinetics, toxicological studies, preclinical, clinical studies and all. So now we'll explain about the regulatory affairs. Then regulatory affairs is a profession discipline consisting of knowledge, interpretation and practical application of the regulations, guidelines, policies and procedures, and operating principles, which is in the manufacturing and the registration, marketing and the maintenance and withdrawal of the drugs. This maintenance is the life cycle. I'll explain further. This RA regulatory affairs role and the purpose. So basically this RA professional, what they'll do, they'll guide for the plant before plant resource, which is a regulatory, R&D development. So in the case of R&D, they'll suggest, so what are the things they can take a chances to develop a formulation. So before initially, I'll start with the when you filing any drug, which is the IND, investigation new drug development. So in investigation new drug development, so it will take around 2 to 5 years. They will be studying of full entire chemical structure of the particular molecule which is going to be established and how it will be and what are the preclinical developments. This is the next stage. So when you are synthesize the chemical product or the chemical bonding with a chemical molecule, and then the next stage is once you finalize with the molecule, then you need to check for them. So what is the actions for that? So how it will affect on the any therapeutic use. So then you are going for the preclinical development. Again, it will take around 2 years approximately. Then you will go for the clinical trials. Once you establish preclinical studies, so based on the data, you can interpret by studying the small populations. Just gradually you can increase how excuse me. So number of patients in the testing of the particular drug. So whereas the preclinical, you are establishing the what is the toxicological dose and the minimal doses and all. When you go for the phase 1 studies, you are checking in the patients how the drug will affect, whether it's intended to the effect which is working or not, the therapeutic area is suitable for that or not. Then the minimum concentration, safety and efficacy studies will do in the phase 1 study. Then phase 2 and the phase 3. Phase 3 is a large study. So phase 2 is the minimum between healthy patients. Then phase 2 is the different sites they will go for the study of this phase 3 study. So once these phase 3 studies are completed, then they will go for submission and approval. Submission and approval process to the regulatory agency. So once the product is approved, then the next stage is you can market that product. So then the next stage, once the product starts in the market, so up to the existing, ending expiry of that product, ending of the supply to that product, you need to maintain all the related records, and so whatever the related information or whatever the queries raised by the authorities, you have to answer that. That is called as life cycle management. See, basically from the establishment, starting from the preclinical, starting from the synthesizing a chemical molecule, so up to the submission process, it will take around 13 to 14 years for the innovator to develop the molecule. Then by that time, only the particular who is registered first, he will be there in the market.
मुझे थोड़ा सा काम है, मैं लेट पे ये कर रही हूँ तेनिसू के लिए सर्फ का पैकेट भी होगा काको.
Sir, you can proceed actually. Hello. Reddy sir? Karna Reddy sir?
Hello.
Yes, proceed sir, proceed.
Yeah, okay, yeah, thank you. So this life cycle management will take around, so maybe it depends on the scope or demand of the particular drug you can able to supply in the market. Until unless which is excluded from the market, you should maintain all the records. So all these responsibilities will be taken care by regulatory authorities only within the company. So once this is the case, when innovator came with the particular drug, then so no one able to can come without. By the time, see what will happen for this taking, he'll take much more time to investigate or innovate the particular. So starting from the single molecule to the final stage of the formulation. So he'll spend so much money and so much of investment of the time as well as so much knowledge he has spent on that. So he need to gain some profits from the market. He will be having only 6 to 7 years. So after the establishing of the particular molecule, see when you go for the patenting or when you have find out in this initial stage, you have the therapeutic properties of that molecule, you will go into the file as a patent. So simultaneously you will be submitting the registration for the application as a what is the condition, what is the purpose of you are doing that. So you need to initiate that. So once you start the submission, file the patents, you will be having 20 years of the time for you to make the profits or the whatever you will be having the royalty period for the 20 years. So out of the 20 years, you will be finishing here only for testing and the efficacies, all those things, and up to the formulation finalization, you will be need around minimum of 10 to 13 years. You need to wait for the market to be product to be delivered into the market. So you will be having only just 6 or 7 years to gain all the profits or whatever the investments you have given spent on that particular development. So that is the reason always innovator products will be having much cost price. So if he spent on the particular thing, if somebody has came once he delivered in the market or whatever the based on the studies which is available in the social media, if anybody comes to the picture and he can also go into the market means, so whatever the time he spent, whatever the knowledge he spent on this, it's a wastage. So for that only Hatch-Waxman Act, he has proposed in 1984, he has proposed a new law. So anyone who can develop particular molecule and anyone can enter into the market in the after 20 years of the lifespan of that particular innovator product. What is his suggestion is anyone who is able to develop or who can compete with that. So no one can sit behind as a simply let him finish, then only we can start. So no one will do that. Simultaneously, based on available information from the social media or within the patents, so like Orange Book, you will be having much information on that particular product. They'll publish. What are the information they'll receive? They'll publish in the Orange Book with the preclinical and the clinical studies as well. So before 6 months, so any one of the who can file, he is the first innovator who can file the immediate next. He can enter into the market along with the innovator product. Along with the innovator product, anyone can come into the market with the same therapeutic he should influence. He should prove that he has not followed any methods for the developing the same criteria or the same method or the same formulation he hasn't used as innovator. He can modify the alternate and he can go into the market. That too, who is the first filer next to the innovator, who will be the successful filing of that particular formulation and he got the approval, he can enter into the market just before 6 months of the expiry of the patent. So that is there we got so many generic players they are having. They'll spend the much more knowledge on. They'll spend much more thing on R&D, research and development to innovate the different formulations apart from or except from innovator formulation. They can go with the same formulation by using different excipients or the different concentrations like that. So this study, Hatch-Waxman has given an opportunity for those generic players. They can rely on preclinical and clinical trials of the innovator studies. So the thing is, once you develop the formulation, what you will do without preclinical and clinical trials, how you know that is equal to the innovator drug? So you have to do BA/BE studies. So with the comparison with the innovator drug, so it's a different scenario when you go into the in vitro in vivo studies and the clinical bioavailability studies. You will come to know whether whatever the reference drug you have taken, where have you meet that particular standards as of a innovator. So again, it will involve a lot of time and investment. So that's why these people have around 6 months of the grace period they can enter into the market. So as such, they cannot enter into the market in this case. So they should meet again approvals from the authorities as well as any objections from the particular innovator. Even authority, whatever the who receives application, they'll send the same information to innovator. So he can suit particular he is having the one of the chance that he can also analyze the things whatever he is filing. he can able to stop the generic manufacturer to come into the market. He can having a chances of gaining much more profits. So this is about drug development. So what is a generic and what is an innovator? How much time it will take? So how generic manufacturer can enter into the market? So once 20 years is done, then everyone they can do enter into the market by continuing all these studies except preclinical and clinical studies. They have to do bioavailability studies, in vitro dissolution studies. So comparing with the innovator product, all these things they have to file, study, and they have to submit into the authority. Then only they can obtain the license. Then they can enter into the market. So whole process will be related on all basically depends on this interaction with the drug authorities to the firms. Within the firms and in the company or the firm or the industry, whatever you call. So there is a department called regulatory affairs. So this regulatory affairs department is a bridge between formulation, industry and to the drug authority. So before entering into the market, so whatever the needs, whatever the requirements for the regulatory, so this regulatory department can guide to the within the industry, and the same thing they can associate or they can associate with the local regulatory bodies or wherever they are going to submit. So they should associate with them, and whatever the queries they will ask, this regulatory department has to answer those queries by interacting or collecting with the information from the different sections of the industry, different departments of the industry. Here, see, company is having a license to sell. Here, our regulatory affairs department is one-stop shop on all regulatory issues. Whatever the queries raised by the authority, if you clear those queries, then only you can able to get the license. So that is the reason I said it is one-stop shop on all regulatory issues. Working towards optimum submissions. Optimum submissions means, I'll explain in the further slides. Yes. So regulatory compliance. Now whatever I said. So when you submit any application to the regulatory authority, definitely you will be having a concerns or queries from the regulatory agency, or they need any clarifications, they'll come to the regulatory. We should be able to answer those questions without any hesitations or without any delays. Those things we should answer. Then risk assessment. So when you got a query, so you have to be in a proactive, and we can suggest which department has to take that particular responsibility, how they have to handle. So what is the perspective on the mind of, since you are interacting with the regulatory bodies, so you can guide how they are expecting the answers. Based on that, you can guide your internal teams, or you can suggest more thoughts. You can share your thoughts. Based on that, internal departments also, that particular respective department, they will come with their answers. So the same thing you will be transferring or in the prescribed formats to the authorities. So if they satisfy, then you will be successor of the particular dosage form will be entered into the market. So it will be boost your company's growth as well. Then market preparation. This is what I am telling you. This market preparation is nothing but you should be prepared, since they will ask some questions. You will be then you need some time to adjust, or you need some time to prepare those things. It will take lengthening of the time. So by the time once you submit the application to the regulatory authority, you should be in a constant communication with those regulatory agencies. Who is you should maintain the particular communication to the proper communication with the regulatory inspectors or the authorities. Then you can speed up the process of what are the queries. You can speed up the process of closing of the queries raised by authorities. See here, good regulatory practices. So any firm, so in case of any firm, whatever the regulatory firm, they should have GMP, good manufacturing practices. Then good manufacturing practices in the sense it comes to the manufacturing section as well as the quality things. Then GLP, good laboratory practices. Whenever you are doing anything in a lab, you should be perfect. Whenever they will come for the inspection, or no need to wait for they have to come and spend on inspection, and they can find out the findings. Instead of that, while working ourselves, we should be having much knowledge and we should be cautious to produce quality documents, quality results without compromising in quality issues. Then, so apart from this, good manufacturing you will do and good laboratory practices. So, how much importance you will give for these two? You need to give importance to good documentation practice as well. Since in the good documentation practice, there is a scientific writing it involves. So you should be cautious when you are preparing any BMRs. So whenever you will go for the GMP or GLP. So GLP is in laboratories. I mean analytical development or the quality control department or in process controls. There you will be having good laboratory practice you have to follow. GMP, good manufacturing practices from starting from API manufacturing to you should concentrate on wherever you are procuring the materials for the preparation of the formulations like excipients, raw materials, solvents. So all those things, those plants also should be having GMP. Then only your product will be GMP compliant. Or else if you are taking any material from the non-GMP plant, if they have mentioned a project, they haven't maintained a proper environment, the thing is what happens, you will procure all the materials. So if it is manufactured in the non-GMP compliance plant, if the same material is you are adding to the all the material whatever you procured except one material which is non-GMP, if you mix those products, who knows, the product will spoil or it will damage to that particular product can damage to the entire formulation, all those things. So where you should maintain the consistency whenever you are procuring anything, you should procure from GMP plants. So this is the practices. I mean GMP and the GLP, those are the analytical and the manufacturing practices. So both the things will relate with the good documentation practice. So here we should be cautious. No errors to be written, no bad dates or those things. When you go into the industry, you will come to know how the industries they are playing and how they are struggling to maintain all these together, how the QA department and how the QC departments or the production departments they are struggling or they are putting their efforts to maintain all into the GMP. I mean good laboratory, good regulatory practices. So once you fulfill all this information and everyone has to rely on their responsibility, then the particular product will come to the market, it will be safe to the patient. So our main motto is to serve the patient. Pharmacist's motto is to serve patients. So you should not be biased in the work. So then only industry or whole world will be in a good environment. If you are biased, then everything is collapsed. If you are not healthy, no one can do anything, right? So you should be always healthy. So by keeping all those in patient in mind, you should work whenever you are working in pharma industry. That is the main motto. Whatever the department you work, you should be focused on patient focus. This was all good practices related to healthcare system. Ultimately, the aim at quality of the product, safety of the product, efficacy of the healthcare products, and thus ensures well-being of the patients. And drug dosage form registration. See, once these things in plant, the manufacturing and quality control and good documentation practice, which is there in the one section. Then, so once everything is established, what is the next step? So everything is established. Then you have to go for submission. Submission of the everything means if you go in the specific areas, then it will take much more time. I'll explain the last. There are few slides also. So based on that, I'll explain in a brief. Then drug and the dosage form registration. Drug means API registration. Dosage form means any of the formulation registrations. See, these are the data requirements for the drug registration. So administrative information, quality, safety and efficacy information. So see, generally if you go in a regulatory department, yeah, I'll explain it later. This administrative information, here you will give who is the manufacturer, where is the origin of the country, and then where is the origin of the supplier who is the developer of the product or the manufacturer of the product. You will give the general packing information, what are the labeling information that will be given in administrative information. Then is quality. Quality information is module 2 and module 3. There you will be having EMC related and the formulation related. Those things you will give chemistry and pharmaceutical data. Here you will cover all the things like quality overall summary. There in a quality overall summary you will give entire sections for from quality, safety and efficacy all together. You will give one section as quality overall summary. And the quality is a module 3. There you will give chemistry and pharmaceutical data in detail. Whereas in you are giving quality overall summary as a brief. In detail you will give chemistry and pharmaceutical data. Here you will give formulation data, chemical data like what are the impurities are there and what is the controls you have given and analytical methods. Then stability studies, stability data. You should give how much time it is stable. Then safety. And in preclinical studies, you can pharmacological, toxicological data. It depends on country to country requirements. This is not mandatory in the US. This is mandatory in UK. So based on the country to country, you need to submit different dossiers. Then efficacy. This is a BAD studies. We will be studying. We will be doing bioavailability studies in the different areas and compile those things, and you have to submit for the along with the all these things you have to submit to the authorities. So these are the data requirements for the drug registrations. So basically we will be having 5 modules in this. This administrative part will come in module 1. Then all together whatever the information we are giving detail here, quality, safety and efficacy, we will give as a one module as quality overall summary, that is module 2. Then module 3. This is the quality part I explained. Then there is the safety part, this is the module 4. And this is module 5, clinical trials data you will give. So this preclinical you can give as a references by taking from the sources, reliable sources like Orange Book, those things they will be having. Those whenever you are going for the patent filing, you should file this. So those subsequent manufacturers next to the innovators, they can take the information from the reliable sources and they can file, submit along with the for all these things to the drug registration process. So once you submit to the authority, then they will give the acknowledgements on the submission. This regulatory department has to maintain all the correspondences from the authority. They should have separate wing and they should maintain each and every communication from the authority until up to you get the approval. Then further you should continue for the up to the product is existing in the market. So there is a registration process. Here I told you types of submissions. If you go for the USA, this will be investigation new drug and NDA, new drug development. This is IND, it's an investigation new drug. Then new drug application. Once you develop, then new drug development will start with the preclinical phase. Then you will file the new drug development. new drug development from the preclinical stage to first innovative product NDA, then abbreviated NDA. First generic product it will come later on the follows. So if you go for the EU or UK, it's a clinical trial application where you are calling it in US it is called investigation new drug application, then marketing authorization application. Then in Canada it's a different, it depends on country to country. Wherever you will go, now it's a global scenario. You no need to go for each and every, each and every reason-wise manufacturing process. That's why RA has having a crucial role in development of these products. Then API, they can go for drug master files. The same way, they have to go for the packing material submissions. What are the excipients you are giving? You should file into the drug master files to the authorities. Then they will give you the grants. There are, even not only in the specific to the formulation and these things. So nowadays you have so many opportunities for the regulatory affairs, not only in the formulation or API. You can go into the specific fields like what is the requirement for the formulation. In the sense you need lot of dyes or excipients. Though they are also trying to comply with the regulatory requirements, and there also we are having the opportunity in the industry. There, that is also one of the major industry. There also you can go and explore your knowledge and you can gain or you can support to the regulatory field. Okay. And the modes of submission. So initially we used to have only paper submission, like they will be having a lot of bundles. It's a time consuming. You need to take each and everything, the printouts. You should bound the things and you have to submit in the individual sections. It's taking much more time. Then later on, once this is establishing, as the time comes, then the next is a mixed submission. Few of the information they can submit to the CDs or like hard disks or however which is the preferable. They used to submit those things. Then the electronic submission. Here electronic submission, they used to send all the informations together in DVD formats. Now e-submissions, they are having e-submission users, they can send directly to the gateway. Submission gateways are there for each and every individual. Most of the established countries, they are following e-submission gateways. They can submit through these gateways all the information instead of sending postal, all those things. They can send all the information into the e-submission gateways. Anyhow, some countries, they are preferring to send at least module 1 in an electronic format through the DVD with the application form. Just with the application form and the DVD of the module 1 they can send to the. This is based on, again, depends on country to country specific regulations. These are the modes of submissions. So these are the different regulatory bodies in worldwide. In India, we have CBSCO is authority. You need to file dossiers whenever you are going to the new drug market. Or in addition to the CDSCO, for the generic markets, we have state level authorities in India. You can submit to the state level authorities and you can gain the license and you can go for market. Then US, it's a food and FDA, and Japan, the Ministry of Health and Labour Welfare. There are the different things like what I have given here. So remember one thing. So technical information is common to almost all the regions. Whenever you are doing any submissions, it will be common, except we will be differing in module 1. This is the administrative data. It will differ to the country to country. There are certain requirements will be based on the country regulations or their jurisdictions. Then the review. Here also we will having nowadays, see how initially when the establishing and the initial stage of the regulatory was there, quality of the application is the main motto of the company or industry, or you will go for agency also. They will be looking in the quality aspect and safety aspect and efficacy aspect application. Whenever you're in a plant, as a business person or as an innovator or a formulator, you have to look into all this aspect as well. In the initial stages, there is a lack of adequate pharmaceutical development information. You are not having much information when this internet and all those things which is not feasible or available to reach and everyone. You will be having there that time. There is the information related, limited information based on that only they have established two things. So based on it was going with a lack of that risk-based assessment, all those things, it was not there in the initial stages. So now the scenario is growing now. So we'll be having different views on review of the application, and by sending of the application also you can send with the, if you have a proper knowledge and the skill set, you can send with the minimum queries. You can expect the minimum queries from the authorities, and you can send the good quality of the dossiers to the authority. There you can speed up the process, you can reduce the time lag for the approval of the drug. In the initial cases, it was the evaluation of anti-CMC section by a single chemistry reviewer, all the CMC parts. CMC parts, as I told you, you will be having manufacturing details and the imperative details and the validation and the stability detail and the formulation aspect, the reference standards, and you will be having packing materials, all the information together. It was reviewing by single reviewer. There may be chances if you have one reviewer focus on all the divisions, there may be chances wherever he is expertise, that case he can focus on particular review as an exception. The remaining he is having idea, but in-depth knowledge, if you have, that's fine. There won't be any issue. If he is having a lack of any idea, if you send the same thing, reviewers, they will be working on different issues, and they will be having a much more focus. Reviewers in the drug regulatory authority, they are well versed on with the reviews. They will receive many of the applications. They will review many aspects. They can directly pinpoint wherever your loopholes are there in the application. So then the next stage of the review, quality assessment. The risk-based assessment will focus on critical quality attributes and the interrelated with the section. Like formulation, you will give the formulation by in some cases you will in some of the applications you will attach some stability data you should give. In some of the dossiers you will give batch records. So existing batch records you should be those information, batch details should be matched with the stability data where you are giving. Those cross-checkings and all, you should be interdiscipline, you should check each and every sections, interrelated sections in the complete dossiers. So when you are reviewing itself, you should be able to predict the quality and the quality of the document, whatever you are submitting. This quality as overall summary. Initially, whenever you submit all the dossiers to the dossier to this authority, they will rely on quality overall summary. So this information will give you the detailed idea about how the entire application is there, whether they have given all the, they have covered all the information is required for the approval of the drug. Then question based review. So generally, if you review, you will go in the general pathway review of based on whatever there in the text, you will review on that perspective. If it is a question based review, then you will be having a chance to think how we have to review. So what is the lacking? What is the information to be there in particular sections? Integration of relevant bioform and microbiological functions into the CMC review. These are the careers in regulatory affairs. So I think all those things I have given in during the course. I'll brief up this. So regulatory specialist and regulatory affairs managers, you can go into the up to the director's level also if you have a sharp skills and the smart skills in the knowledge. Compliance specialist in the SME. That's what I am telling. Whenever the review based mode, compliance specialist, whenever you receive any query, you should be able to answer and you should first you need is first and the foremost quality you need it. You need to understand exact what is the question they have asked in the which prospect they have asked regulatory authorities. Then only you can give exact answer to. You can guide the team within your departments and within the plant. You can guide the different departments and you can get the information from the plants and you can satisfy the regulatory authority without expecting further queries from the same. That's what main motto here to tell as a compliance specialist. Then SMEs. SMEs means subject matter experts. Based on your interest, you can choose any of the field, like you can choose quality, you can choose the production managers, or you can start your career in the quality control or quality assurance, or to the production division, or to the packing division, or you can go into the clinical or non-clinical, or you will go to, as I told you, you can go into the dyes manufacturers, or you can go to API. Based on your interest, if you have thorough knowledge in the interest research chemistry, you can go to the R&D section. You are well versed with the analytical knowledge. Based on your skills, you can set your goal and you can move to the different fields on the starting of your career. Don't think regulatory affairs or the regulatory department is the one you need to get directly first as the first job of your career. The main, whatever I said here, this compliance specialist, you need to have different knowledges and different departments of the knowledge, different areas of knowledge like chemistry, manufacturing, and the safety. All those things you need to have. You can start with any career with any of the different different fields. Then you can jump into the, if you are really interested into move to regulatory, simultaneously when you start with your career in any different field, you try for the regulatory affairs. If you are not go through into that field, you can start with the other, like you can go for the manufacturing or any other field. The thing is, you should don't lose the scope or goal to move to the particular department. For that, what is you need to do? You should have a background. You can continuously search and follow your guidelines, or you can associate with any other journalists. I'll go through those things in a minute. So if you have a complete knowledge on those things, you can start with. If you are much aware on all regulations, global regulations, you can start your own consultancy also to assist the clients in the regulatory submissions. These are the educational requirements. Any undergraduate degree or disciplines of the life sciences or the clinical sciences, public health, you can go to this field. The main thing is you need to have proper communication skills if you want to elaborate or if you want to establish in a regulatory field. Proper communications means it's not only just you are well versed in the talking of the English. So whatever the other person is telling, you should understand and you should deliver the same thing to your colleagues or the superiors. You should advise to the different divisions. Then only that is the meaning of proper communication. So don't feel that when you are talking only in English in a good manner, it's not only says to the communication. The way how you are representing or how you are taking the problem and how you are giving the solution. Yes, regulatory professionals began working in different industry before they are transforming the regulatory affairs, as I said before, according to the RAPS report. In this case, if you want to, from the undergraduate, if you can go to the regulatory field also, then you can have, if you have any additional courses on the regulatory affairs field, you can certify this RAPS certificate. There is a website. You can note down this RAPS certificate. You can check in the website. If you have that certificate, you will be industries will prefer you as a preference. They'll give you the preference for you first. Those who are having the RAP, then if you are 10 people are attending to the interview. If you're having like this certification, like RAPs or any additional knowledges, if you are expertise in any of the field, then they'll give a chance to you in among those 10 people. This cross-functional trainings like science, pharmacy, nursing, great scope anywhere in the world, not only in India. This is the thing, if you want to be elaborate or if you want to excel in your regulatory field, you just try to always have a look on guidelines. Always you should be in touch with the guidelines wherever your region you are working or if you are the local regulatory. If you have those things, you can establish yourself like anything. Even now, initially they'll be having only few things, and they are based on the knowledge and the resource. In companies, they are preferring to select the candidates. Now, even employers, since the internet is available to each and every person, even employers' expectations also now they have increased their expectations. They don't want to go for the basic knowledge you are having. They need the multiple disciplines. I mean, multiple exposures at least you have, multiple knowledge at least you have. If you don't have exposure also, at least you are able to understand what they are telling. You should have at least minimum knowledge on different scenarios. That's what I am telling here. Most prospective employees are more common. Finding someone who is possesses the advantage in regulatory knowledge on hands-on experiences. That's what I said here. How to start your career opening is a much or struggling to find the right level of combination of skills fulfill their needs. Stay current in the sense you should be stay up to date with the guidelines. So those things and subscribe to the updates from the publications or the journals. Sharpen your skills. Regulatory projects are usually complex and the time constraint. Cross-function, it relates to the cross-functional as I told you, CMC, all those things, some modules will be relevant to the other modules, there you should correlate. So the thing is you have quality overall summary. The entire application is having the different modules will have different sections in the detail. You should summarize and you should collate the information in one section. So for that you need to having a sound knowledge in cross-functional areas. Then you should be proactive. Proactive in the sense, this is what I am saying. When you submit any application to the authority, background also you can have a free time, you can go ahead with one round of review. Then you can find out, you can expect the questions what they will ask. At least minimum. Fine, you can say that we are thorough reviewing, we are sending that application to the authority, then how you can expect the questions. Then why most of the companies they are facing the issues to answer the compliance reports. Most of the companies, whenever you file, they are getting the questions. For that reason, you should be prepared. Once you submit, don't leave as such and wait for the response from the authority. You just have a look, have a team. Then if you have a sound knowledge, then you can go into the different exposures. You can review those once again. At least you can expect minimum of the query. By the way, you can prepare in the back end. Once the query comes, you can attend easily those things. Even those queries, the reason why I am telling you, those queries, whatever the questions asked by authorities, they will have a specific time limit. For that, you should be prepared. Once it comes, if you prepare, sometimes fine. If you understand clearly, if you have back knowledge, some knowledge and some background evidence, you can answer the query easily. If you have sufficient data, you can answer it easily within the time frame. Or else you may lose the opportunity. Sometimes if you are not answering that particular question, there may be a chance of rejection of the submission dossiers also. So in order to avoid all these things, you should have proper communication and the proper knowledge and sound knowledge on the wherever you are expert, wherever you are having knowledge, you feel that there you can explain your things. You can excel your knowledge in the in-depth knowledge. You just gain the in-depth knowledge in that particular field at least. That will be helpful for you. this is the thing. Now the whole regulations they are harmonizing. Initially it was an individual country to country it was there. So now most of the things are harmonizing. This harmonization has started with this harmonization is started with ICH guidelines. Initially these three countries which were there in ICH formulation, now also these three are the ICH countries, Europe, United States and Japan. They three, they have formed as a unit and they framed a few guidelines like quality, safety, efficacy and miscellaneous guidelines. If you go to ICH website, you are having a lot of information. So these are the current trends in the regulatory affairs. CMC authoring, this is there from the starting when you are regulatory affairs as CMC authoring as it was the initial. This life cycle management, the thing is life cycle is having a much scope in future in the sense from the starting 2021, all the patents are going to be expired. There will be a lot of scope for this life cycle management. You can go with the different formulations. You can go with the different or you can go ahead with the you can 1 second. Over here. Life cycle management, and the ECTD and the publishing and the project management. So nowadays, initially you can with a certain scope for the regulatory affairs. Now, just the past 10 years, we are having in India also there is, since the labor cost is less, most of the companies they are migrating to the India. So they can get the better resource, the minimum pay they'll get by comparing to the foreign. Further, the reason they are focusing on India to establish CROs here and ECT in the publishing. This is one of the most suitable job for the those who are having the minimum knowledge also. ECT or the publishing. This is the ones the regulatory documentation is prepared. We have set of the documentation is fine. Then initially how we are doing is we used to submit as a paper or through the CDs or the hard copy. So nowadays the CPD is coming in an electronic CPD. That is once you have set up with all the documents, then you should compile this into the different tools you can use for the publishing. When it comes to the industry, you will hands-on experience only you will gain this. So initial starters you can get with the basic knowledge whatever you are posting or whatever you are studying. You have basic knowledge on the guidelines. In addition to the academic curriculum, you should expose yourselves to minimum of the guidelines and the authorities, what are the regulatory bodies are there. Nowadays research are much easier. Starting from the final year of your degree, if you focus on this particular things and spend some time on the guidelines, then it's an easy cakewalk for you to go through the regulatory field as well as to the moving to the CROs. Yeah, since if the CROs are establishing here, we are having much more chances of getting the jobs here for the pharmacists or the pharmacy those who are having a license background. The next thing is once you are having the knowledge or gaining the experience, then you can move into the project management also. Project management, the CROs are focusing on basically they'll having a different sections. They'll establish different sections in the project management. Why this project management they have brought up this into the field in the sense they should minimize the time to response to clients. So whatever the communications it will be done with whatever the requirements for the clients, we should be taking care on the agreements, whatever those things will be handled by different departments, and same work, whatever the authoring, whatever the queries, those things will be handled by regulator. So that is the reason they have developed project management. If you have a knowledge on the regulatory aspects, then you can aggressively communicate or you can actively participate in the high level meetings. That's the reason if you have a basic knowledge, basic idea, basic experiences, keep on growing, you can have different scenarios or the different exposures, different levels of hierarchies are there. Even your hierarchy also different fields you can elaborate based on your interest. Then social media, yeah, that is the thing one now. Most of the companies they are coming into the social media also, like you see online pharmacies. So at least if you are interested in that, at least you have some knowledge on that also. How to present your product in social media, how you can attract the person, those who you having a number of social platforms. So how can you give your that is a major thing. How you represent focus, that is a major thing. They are looking for such type of knowledge also. I mean the thing is it should not be in the single way, right? So this is what all those things I covered in my topic. This is for the schedule and for the India. So all these topics will be, if you focus on one, at least you will gain a knowledge on how the industry and what is the structures will be there in the industry. How before entering into the industry, at least you have some knowledge on schedule and you can, it will be easy for you to adjust to the environment. This is what the different guidelines, stability guidelines in the ICH has established the different areas of guidelines like I said, the quality, safety, efficacy, and the miscellaneous guidelines. So these things are the stability guidelines. If you go through the ICH guideline, you will come to know for the analytical and the impurity level. These are the different type of pharmacopoeias. Q5 is for the biotechnology and the Q6 is for the specifications. These are the good manufacturing practices. So after basic development, then after that they have feel that these also should be added to the guidelines. Quality risk management. This is the thing how once you establish initial, they have established the few guidelines. Once upon the reviewing of the dossiers or the concerns with the clients, these regulatory bodies they have formed and they have suggested these guidelines also to be included in the field of pharma field. So now this is a continuous process based on the queries which are coming from the during the development stage. If you notice to the regulatory authority, you are facing such type of issue, then they will form and they will see the opportunities and they will take the public opinions. Then they will form a draft guidelines. Then they will post into the websites based on, again they will take the back ends of their opinions and the experts' opinions. If everything is fine, they will go ahead and defining new guidelines which is relevant or required for further expansion in the regulatory field. Yeah, that's what I said. This is the basic knowledge you need in the conclusion. Agencies and activities restructuring the departments to improve use of science and pharma industry, improve openness and transparency is a much. If you are open and transparent, then you are limiting. Sky is your limit to expand yourself. Even the regulatory bodies, they are also aiming to be in a transparency. New and developing is becoming increasing regulated demands for the additional specialized regulatory constantly. So graduates from the scientific backgrounds who enjoy the communication and the teamwork, they like with the multitasking. Those who are the multitaskers, they can choose this as a career goal. Yeah, that's it, Karthikeyan. Anything
Yes, sir.
Hello.
Yes, sir. Yes.
Yeah, Karthik.
Yes, sir. Yes, sir. Thank you so much, sir. And yeah, I request all the participants, those who are in the Zoom platform, to put their queries and questions in the chat box. And those who are in the YouTube, those who are watching that of your live session, the YouTube, we request all the participants put in the YouTube chat box. So we'll be representing the queries or question to the top you, Dr. Reddy, sir. So there is a query from the participants.
Yeah, Karthik.
Yes, sir. Yeah. So there is a query from the Priyanka Savant participants. The new drug require around 10 to 15 years to come into the WFM market. So what about the safety of the new drug in the COVID-19 vaccine, sir?
See, in the COVID-19 vaccine, how they are doing that, they might be skipping of some studies based on the major focus. They'll go for the basic knowledge. So this is the emergency case. So you no need to wait for the until and unless 15-20 years. That is the case. Every entire population will be out of scope. That's why whatever the available drugs, like you see paracetamol or some retroviral drugs which are already there, they are trying with the different established drugs only. They are again rechecking those whether they'll affect on that. That is a one way of minimizing. And with the basic having any idea on that, I mean basic therapeutic area in the sense, if it is continue to affect effective, then they are going for the studies. I mean, the therapeutic index is good, then they are going for the improvisation of this vaccine.
Yes, sir. Thank you, sir. There is another question from Bipanviti Satpati. So, sir, the clinical development of the drugs takes 6 to 7 years. Then how the vaccine come within 7 months?
Yeah, that's what even I expected these questions.
No,
So
no,
this
actually
is
there
a
is
different
a query
scenario.
because
See, yeah,
yeah,
that's
I
what.
would like to represent the question by question. That's only.
Yeah, I got you. I got copy. This is a
Yeah,
different. That's what I am telling. Even before I said, initially we went with the supplements. What are the supplements with the different drugs? Even you know that this is an
yeah,
unexpected disease. It came. No one has sit behind and drug can develop. Then only you can go for
yes,
the treatment. This is not the case. Like they went with the whatever the existing drugs. They went to the directly. They haven't come in a vaccine trial. I mean vaccination. They were
yes,
tried with this different type of medicine which is already available, like antiretrovirals. Based on that inhibitions, they went with. Based on that inhibition information, they went. Anyhow, they have structure. Based on that, they have at least. I mean, they might be having some therapeutic effects of that particular vaccines. Those who are developing. Based on that information emergency, they are going in to to the. I mean, they are conducting all the studies right just with the therapeutic effect. Then they are going for the whatever I said, clinical
yes, sir.
trials. With the,
Yes,
now,
sir.
see, initially it was taking that much time. Nowadays, with the vast availability of the knowledge as well as the information as well as the equipment availability, they can screen together at a time. Initially, you need to wait the 1 month or the 2 months to screen the particular samples. Now, within a 24 hours with the sophisticated instruments, their screening is easy nowadays. That after the screening, they are coming to the next levels. So they are doing the trials, even clinical trials. They haven't come right. It's almost 1 year. Even now they are in the clinical stage, right?
Yes, sir.
Yeah. By that,
Yes, sir.
that's
Thank you,
the
sir.
reason. Since the
Yeah,
emergency case,
yeah.
you need nothing. No one can help in this.
Yes, sir. Yes, sir.
With the
So thank you,
minimum
sir.
trust, you should go.
Yes.
Yeah.
So thank you, sir. There is another question from participant Rumisa. What
Hmm.
are the policies for the post-marketing surveillance? What are the policies for the post-marketing surveillance?
Yeah. Once you, I mean, ADR, adverse drug reactions to be filed onto the authorities. If you have any, so once you expose all the things you have studied, if any patient is having any no, I mean any damage together, if they report, then you have to file the those things or the studies to be within the 15 days to the particular regulatory authority. This is what happened. You have to clear the why it has happened, whether it's a mistake from the patient or is there any chances of the in the development or the particular stability area, particular data, it's happened. So you should investigate and you should file the things or the to the particular regulatory authority within 15 days. That is the pharmacovigilance department. They'll take care on this.
Yes, sir. Thank you. Sir, there is another question from Kalim Sheikh. Sir, after the diploma in pharmacy, can I get a job in that of your regulatory affairs?
No, that's what I said. The diploma in a pharmacy, you cannot get a job. So at least you should have minimum life science background. At least you should have undergraduate.
Okay. Thank you, sir.
So
So
diploma
another
in
thing.
pharmacy, see, the thing is,
Yes, sir. Yes.
regulatory affairs department is not only in the industry. If you have a good communication skills or the knowledge, you can establish yourself into the representative, medical representative. It's not a small one. It's a big, huge scope for medical representatives also.
Yes, sir. Yes, sir.
Yeah.
Thank you, sir. There is another Vaishali Chaman participant is asking another question. What is the scope of regulatory affairs in outside India?
See, wherever for the regulatory affairs, only the criteria is you should have proper communication and transparency in your and with sound, I mean knowledge and you should have a hand in guidelines. If you have a hands in your if you have guidelines in your hands, you can wherever you have much more you can scope establish yourself. Not only in India. That's what I have given in one of the slides. Just 1 second.
Yes, sir.
Yeah, you can
So
establish
thank you, sir.
anything. See, if you have
Yeah.
much knowledge, you can establish as a consultant also. You can go ahead.
Yes, sir. Sir, there is another question from yeah.
One second topic. It's a global scenario. You can anywhere you can establish yourself in regulatory field.
Yes, sir. Yes, sir. Yes, thank you. Thank you, sir. That is from Srayesh Desai. So, what will be the salary structure for the regulatory affairs job, sir, for the starting careers they're asking?
Starting careers, it's again it depends. If you go to CRO, that is a different case. If you go to the formulation, I mean, in a formulation plant, that is a different case. See, in the CROs, they'll serving for the multinationals, and they'll be having clients are from abroad. Their structure is different, and whatever the Indian pharmacy we are getting is a different scenario. So, if you go into the top level, in the high level case, if you have a basic knowledge in the formulation or you will be exceeded. Okay, you cannot imagine that as I am getting after 20 years. If you are staying in a formulation or in a plant or any plant, you cannot imagine that no one can get that much. This all depends on your skills and the knowledge, the way you are representing those things. It's
Thank you,
different.
sir. Thank you so much.
Yeah, that is a
Yeah.
basic.
Yes, sir. Thank you so much, sir. Sir, there is a personal question from my side. Actually, as we know that there is a industrial job opening, where to find that actually? Where we are to find that this opening in that of industry? How to know that actually? Because most of them is not aware about how to. Because medical representatives only the jobs is they are publishing the ads in that of your any of the newspapers. Let me know how to know the vacancies in that of your regulatory affairs. Where to find that this kind of openings. Can you please guide us actually?
See, nowadays, see, Karthik, I'll tell you frankly, these openings and all, nowadays, the structure is different. They are posting in several social media, even Twitter also you are getting, and even Naukri.com, if you post there, search there you are getting. The main thing is you should build your network. See, you are in a third year or the fourth year in your graduation. You should maintain proper communication with your seniors. Within the observance, within the reference, they are absorbing the resources. So now the scenario is changed. Few of them are coming from the portals, few of them coming from the internal end. So that's why you should maintain a proper communication with your seniors as well as the new person. 1 second. See, you just here I given regulatory affairs professional society. You just register in this. the organizations for the professions and the regulatory affairs. In the slide, Karthik, here I have given. Hello?
Yes, sir. Yes, sir.
Yeah, if you register,
Yes, sir.
in,
Yes, sir. Yeah.
yeah, if you register in this, there is a lot of aspirants will be there. If you go into the LinkedIn also, there is a lot of, you select that field and you can touch on that. You will having a lot of members will be there. You just
Okay.
talk with that, and if you are matching with anyone, you can continue journey with them. Then it will be a chances for you to move into that. Then you can guide your next. There is a chain thing now.
Yes, sir. Yes, sir. Thank you, sir. Thank you so much. Now, I would like to conclude the session actually because all the sessions are over now.
Yeah,
Now, yes, sir. Yeah.
right, so thank you very much.
Yes, sir. Yes. Yes, sir. So, sir, I would like to conclude the session. So, Dr. Karna Reddy, sir, has elaborately and share his experience in that of your various opportunities in the industry, and he is related to that of your. He briefed about what is regulatory affairs and roles and purpose of that of your regulatory affairs, and what is the job opportunities in that of you, and also what is the Hatch-Watch Act 1984. And he has explained that of your company how to make RA crucial for the company's success and regulate that good manufacturing practice. He explained detailedly, and also the dose and dosage form registrations, and also types of submissions for the various regulation depending upon the various regulations of outside India as well as inside, and also CMC review practices. Also, he explained very detailedly, and pharmaceutical quality management assessment, and how to start your career in the regulatory affairs. So there is a thing SR has covered very broadly actually. So I think this will be useful for that of you those who want an opening in that of your industry, particularly those who are interested in the regulatory affairs, or maybe after the PG. Those who are studying Sutix or any pharmacy, they are interested in the regulatory. So it will be very useful for them to make and get an opening into the regulatory affairs. So I also shown the slide now at that is how to get a job into that. So one thing is you can be in touch with that of your regulatory affairs professional society, or you can be in that of your the organization of professionals, the regulatory affairs, as well as you can be in that of LinkedIn. LinkedIn, I will be getting a lot many people those who are working the regulatory affairs. You can be in touch, and as I told that you can follow them, and if you got a good opening, it will be just good for you. So try to follow that as the instructions are as given, and also the current trends in that of your regulatory affairs also is covered very elaborately, and RA knowledge and the skills, and Schedule M for the for India, and international counts and ICH guidelines he has covered very detailedly, and also the stability and analytical and impurities testing and everything, pharmacopoeias and quality of biotechnological products, specialization, good manufacturing practice. So this CSR has covered as Dr. Karna Reddy is having more than 10 years of experience in that of your regulatory affairs. So she has shared their experience very minimum, and he has explained that of you what are the different job openings in that of your regulatory affair. I am thankful to you, sir, on behalf of that of your Sydus Publishing, Kerala, India, and also that of your Kalinga Biomedical Research and Training Establishment, Odisha. I would like to thank you. Thank you so much for your sharing your knowledge and orders. Thank you, sir. Thank you so much, sir. Thank you.
Thank you.
Sir, you can stop sharing your slide. Thank you.
Thank you, Colonel Reddy sir, for your informative session. Next, we'll move to today's second scientific session.
Sir, can you please stop sharing the slide?
Yeah, that's what. How I'm not able to.
Just you stop it, sir. That's all. Stop sharing. That's all.
Okay.
In the red button. Yeah. Thank you. Thank you. That's all. Thank you. Thank you so much, sir. Thank you so much.
Yeah, thank you.
Now we have Dr. R. Nirmala Devi, Assistant Professor, Department of Biochemistry, Avanaslinga Institute of Home Science and Higher Education for Women, Coimbatore, Tamil Nadu, to deliver the topic on advanced technique in bioscience research. I would like to invite Dr. Karthikeyan, convener of this event, to introduce ma'am. Please, sir.
So today is the second scientific session and the conference fourth scientific session. The topic on advanced technique in bioscience research is going to be delivered by Dr. Nirmala Devi, Assistant Professor, Department of Biochemistry, Avanashlingam Institute for Home Science and Higher Education for Women, Coimbatore, Tamil Nadu. I would like to introduce Madam, that is Dr. R. Nirmala Devi. Madam is currently working as an Assistant Professor in Biochemistry Department in Avanashlingam University, Coimbatore, Tamil Nadu. She is having more than 20 years of teaching and research experience, a specialization in the medicinal plant research in leukemia and lithiasis. She has guided 6 MPhil students and 3 PhD students, and 3 PhD students has been awarded, and he is currently guiding 4 PhD students. She has published 21 international journals and 5 papers in the national journals. She has received more than 20,000 grant from the Avanashlingam University and rupees 1.4 crore rupees from the DST. She has organized 9 seminars, conference and workshops. She was attended 29 seminars and conference and workshops. She has delivered 6 invited talks. She has visited abroad Malaysia and Indonesia for his research collaboration. She is an in charge for Advanced Research Laboratory. She is a staff in charge in Association of Biochemistry, Biotechnology and Bioinformatics from 2007 to 2019. He is a member secretary of Institutional Ethical Committee. She has published 1 book and 2 book chapters. And she is a member of professional bodies like Society for Biological Chemists, life member of Indian Academy of Science, annual member of National Science Congress, India life member of, she is received NSS Best Program Officer Award for the year 2013-14. She has been invited to attend the meetings of the course writers for the course 1, Basics of Life Sciences, for the modular program in Bioinformatics on August 6, 2010 in the Indira Gandhi National Open University, New Delhi. And he has invited and meeting as a course writer for the course 3 and 4, the Basic of Life Sciences, for the module program in the Bioinformatics 26th and 27th October 2020 at the Center of Bioinformatics, University of Kerala, Trivandrum. He is an NSS program officer from 2012 to 2015. He is the Youth Red Congress program officer for the academic year 2012 to 2015. He is a staff in charge for the CSS of the postgraduate student for the department of 2017 to 2019. With this brief introduction, I would like to invite Dr. R. Nirmala Devi, Madam, to deliver the talk. Madam, please.
Thank you, sir.
Yes, ma'am, you can proceed.
Very good morning to all the participants of the national level online conference on innovations in pharmaceutical and biological sciences. I take this opportunity to thank Dr. Karthikeyan, editor, Sydus Publishing, for having given me this opportunity to share my views on advanced researches in biological sciences. Thank you, sir. Am I sufficiently audible to all the participants?
Yes, madam. Yes, madam. You can proceed. Okay. You can share your screen.
Shall I? Yeah.
Yeah, please.
Is it done?
It is not visible actually. Can you make it? Yeah, no, okay. Make it in the slideshow mode. Slideshow mode.
Yeah, I already made it. I think it's taking some time.
Maybe that due to in the upper way. Yeah, editing. It's okay, fine.
Yeah. Okay. So,
okay, ma'am, you can proceed.
dear participants, you could have listened to the previous lecture. I would say rather that lecture was at the end of the drug discovery process, and this deliberations of this particular lecture would be the initial process of this drug discovery. So it will be rather form the basis of this drug discovery. So in the previous lecture, you were listening after a drug has been discovered, what are the procedures or the regulatory efforts that need to be followed at the end in order to make it commercially available in the market. But this lecture forms the outline of before discovering a particular drug, what are the essential laboratory skills or the research skills every researcher must possess. So this lecture finally outlines what are all the advanced techniques that are essentially required to go for a drug discovery process. So with a
Madam,
brief
please can you
introduction, I would say,
unmute? Madam, please unmute your video also, please, let the audience.
okay.
Video, madam, video only.
Yes, I'll do it.
Your video only, actually. Yeah. You can see in the bottom, madam. We can.
Wait, sir, wait.
Please, slideshow is slideshow is ended actually. Yeah.
Should I unmute video? It's already unmuted.
Yeah, unmute, madam. Unmute it actually. Let it be. Let the audience see you also. Yeah. Yes, better. Yeah. And share your.
Okay?
Yeah. Yes. Okay, madam. Yeah. Madam, share your slides. No. Okay.
Okay, sir. Is it okay?
Yes, yes, madam.
Coming.
Just a minute.
Okay.
Take it.
I'll just make it slideshow. Is it fine,
Actually,
sir?
the slide is not visible actually. No, madam, slide is not visible actually.
It's not visible. Then I have to mute my video.
No, muting is not a problem, madam.
Again,
Actually, you have to open your slide, then you have to share it actually. Open and keep it in mind actually. Open and keep it the slide. Then just you share it. Then you can see the slide. Then you can click it on that. If you go directly open into any of the folder, it won't be visible, madam. Ma'am, you are muted actually. You are muted actually.
Voice.
You are muted, madam. Actually,
Voice is muted.
your voice is muted actually.
Now.
Yeah. Okay. Now it's okay. Okay, fine. Then share your slide, madam. Your slide is not visible. Yeah.
Okay, sir.
Yeah. Okay. Make it in the slideshow mode, madam. That's all. Fine.
Okay.
Slideshow mode.
Is it fine?
Yeah, it's fine. Thank you. You can proceed. You can proceed, madam.
Okay, sir.
Yeah.
So the main deliberations of this particular lecture include the advanced analytical procedures that are used to determine the purity, safety, and quality of drugs and chemicals. And these pharmaceutical analysis rely definitely both on the qualitative and the quantitative chemical analysis that ensure the raw material that in which we are interested have to be used to meet all the desired specification, and also to check the quality of the final product that we are going to obtain as a drug to treat a particular disease or disorder. So the examination of such raw material is carried out to ensure that there is no unwanted or undesired substance that is present which might deteriorate the manufacturing process or appear as a harmful impurity in the final product. So my deliberations would have an outline of various techniques, including flow cytometry, two-dimensional gel electrophoresis, and under chromatographic methods HPTLC, HPLC, GCMS, LCMS, and imaging technique with confocal microscopy, microarrays, and RT-PCR. So the participants need not worry about the slideshow, which is running more than 100 slides, because many different techniques I'm going to discuss. All images I have incorporated in my slides just to make the deliberations very clear. Okay, so to begin with, we'll just start with flow cytometry, which is a very significant and crucial instrumentation in any of the drug discovery aspects. And why are we starting with such significant flow cytometric invention is, which finds to be a bottleneck discovery in drug processing and development. So if you see, if you coming to the applications of this flow cytometry, enormous applications starting from immunogenicity testing, pharmacokinetic analysis, antimicrobial discovery, HIV therapeutics, screening bioactive genes, biomarker discovery, small molecule discovery, drug action, drug safety assessment, and finally drug efficacy. All these can very well be assessed using this particular instrument, flow cytometry. So coming to the principles of this flow cytometry, this particular technique is used to detect and measure physical and chemical characteristics of a population of cells. It is like rather cells, you can analyze even any other kind of particle thing can also be analyzed using this particular technique, but essentially the characteristic features of any single cell can be obtained using this flow cytometric technique. And apart from this single cell characterization, cell sorting, physically separation and physical separation of the cells and purification or also can be obtained based on the optical properties of this flow cytometer. And coming to the instrumentation part, this is the flow chamber through which the cells or the cell suspension would be passed, and it will be interrogated with the laser scheme, and this forms the optic system where you can find the forward scattering. The light which is interrogating with that will be scattered at 2 points, form a forward scatter light and side scatter light, and this forms the basis. And this forward scatter light would be giving the characteristic size of the cells, and this side scattering property of the scattered light would help us to derive the granularity property of the cell. So that is how these are the various places where it will make us possible to understand the characteristic feature of the cell, and later this will be detected, and this optic signals will be converted into an electronic signals. So that is why what can a flow cytometer tell us about a cell? It can give you the relative size of a particular cell, which we are interested in analyzing, and also the relative granularity or the internal complexity of the cell can be analyzed based on the side scattering and forward scattering matter that we are received from the optics. This relative fluorescent intensity marker can also be very well analyzed if it is tagged with a fluorochrome or a fluorescent intensity. So this schematic representation will help you to analyze. So this is the light source or the laser beam with which the incidental interrogation happening, and that leads to the formation of the forward scatter and side scatter. Okay, so these two scatter helps in interpreting the results like this. So if you take a blood population which will be having many subpopulation of cells including basophils, eosinophils, neutrophils, erythrocytes, lymphocytes, lymphoblasts, and whatnot. So these many different types of cells can very well be segregated and identified. Their characteristics would be identified like a scatter plot what is displayed here. So from the forward scatter plot, you can identify the lymphocytes, and from the side scatter, it is possible to differentiate neutrophils and monocytes in this case. So this is how the scatter plot or the cytogram would appear at the end of the experiment in flow cytometer. And these are the various specimens or the samples that can be assessed using flow cytometer. So any type, it may be a fresh blood sample or tissues or lymph node, but after processing the sample, this can very well be utilized for assessment in flow cytometer. and this is the instrumentation aspect of flow cytometer. As these are the major principal components that is built in the flow cytometer. So as everybody, all of us know, spectroscopic methods or any other spectrophotometric methods which uses the light source, and it will be detected whatever the signals obtained from the after interrogation of the sample, it will be detected. Okay, so this comprises of fluidics component, optics component, and electronic component. So this fluidics component is nothing but focusing the cell suspension to the laser beam, and once when the laser beam and the cells are interrogated here, so you can see this is the laser beam, and here the cell suspension is coming. So when these two are interrogating at a point, it will be scattered. The light will be scattered, forming the forward and side scatter. So that portion forms the optics, and whatever the optical signal that we receive, that is forward scatter or side scatter, will be converted to a digital signal, and that will appear as a histogram or cytogram. Okay, so here again, how this is possible, and in the main principle I was telling, a single cell can be characterized. So but when your suspension forms to be a cell suspension, there will be a mixture of cells present in it. But what is the main advantage of this flow cytometer is this can analyze a single cell at a particular point. And how this is achieved, that is because of this hydrodynamic focusing, and this is achieved with the help of the sheath fluid. So a large number of sheath fluid will be forced through a compressor, and because of this hydrodynamic focusing, the entire cell suspension will be converted to a single target cell, and which will be interrogating with a laser beam, forming the forward and side scatters. Okay, and this is the fluidic portion. As I was telling, sheath fluid is driven through the flow chamber by air pressure supplied by a compressor, and then all that process will be happening in order to target a single cell at the focus point, making it what is called hydrodynamic focusing. And these are the various fluorochromes. Only these fluorochromes, when attached to the sample, help us to make the optics point of the flow cytometry very clear. So some of the fluorochromes that we use here helps to specifically target any cellular markers or antigens that are present in the cell suspension or the individual cells. And this is, everybody knows about this principle of fluorescence, with which a particular wavelength, this fluorochromes will be excited, and this will be emitted at some other different wavelength, and which will be taken care. So this is the excitation spectra. In particular with flow cytometry, these are the important fluorochromes very frequently we use in the technique. So FITC, PE, PERCP, and APC. So very few, very important ones I have taken. So you can observe all these 4 different fluorochromes are having a specific emission excitation wavelength, and after interrogating with the laser beam, that will be emitted at a different wavelength. So that is what will be defined with the help of the detector after passing through filters. Okay, so this is what in the previous slide it is like, it is given 488. For instance, if you see 488 is for FITC. What is the wavelength for FITC is 488. So this is what I am giving in the form of table here. So these are the various fluorochromes, and based on the sample, based on the nature of the sample to be determined or analyzed, we can choose the fluorochrome. There are several many fluorochromes are available. Even combined fluorochromes will give you a better result. And the optic system, not only the laser interrogation is happening, after that, as I said, forward and reverse side scattering will be happening, and all that light signals will be screened through the optical and beam splitters. Only then the signal will be taken to the detectors. Okay, so this is the light path. After passing it, you will get forward scatter and side scatter. These scattered lights will be captured in the form of light signals, which has to obviously, so this will, as already I've told, forward scattering correlates with the cell size. So depending upon the size of the cell, there is an increase or decrease in the forward scatter would be observed, and depending upon the granularity nature of the cell, there will be increase or decrease in the side scattering. So this slide will give you an just idea how these laser emissions. I said the fluorochromes will excite at one particular wavelength, and it will emit at a different wavelength, and it will be passed through some of the filters like band pass filters, long pass filters. All these filters it will pass through, and then finally it will be detected. So that is what is given in the entire thing. So this is the laser beam where it is getting, and from this flow chamber, the cells are exposed to the laser beam, and all the lights are passed through these filters, and finally it will be detected by a detector to get a cytogram. Okay, so these are the various filters. I am just skipping just to get a specific wavelength, because the fluorochromes will excite and emit at a particular wavelength which have to be captured properly, for which all these filters are used. Okay, these are the various filters that we will be using, long pass filters. So the main significance of this is wavelengths greater than 500 nanometer will be captured by this. Like that, each filter has its own property. Wavelength less than 600 nanometer will be captured using short pass filter, band pass filter, dichroic filters. All these are the various filters just to make the signals prompted correctly to the electronic signal. Okay, so now you see from the flow cell, all this is passing through that, and finally it is detected by the detector called photomultiplier tube. So coming to the detectors are of two kinds. One is photodiode detector or photomultiplier tubes. So whatever the signals that are received from the optics will be received by the photomultiplier tube, which will enable the conversion, digital conversion. Okay, so here comes the electronic system. So whatever the light signal that are received after interrogating the sample with the laser beam, all that have to be converted into an electronic signal, for which this electronic system is very much helpful, and this will be finally forming what is called cytograms. So this cytograms, either histograms or cytograms will be formed. So this correlated parameter means then it is nothing but cytograms, and this cytograms may display data either in the form of a dot plot or a contour plot or a density plot. So this is the histogram. You can see this is how the histogram of a flow cytometer appears, and this is the cytogram of the using this is PlotStat is the software that is built in with the flow cytometer instrument, and therefore using any of these software you can have either histograms or cytograms as the interpretation of the technique or the experiment that you perform. So I said the cytograms may be a dot plot or a density plot or a contour plot, and this is an histogram, and this is the dot plot, and this is the density plot. And here you can find this is one population of cell, and this is another population of cell, and these are segregated. So when you are passing a cell suspension through the flow chamber, at a particular point a single cell is interrogated, and after which, based on the program set, based on the forward scattering and the side scattering, the population of the cells will be segregated like this. So when you send a mixture of cells in a population, finally you will get two different populations segregated like this in a dot plot, and this is the contour plot. So how do you interpret this? Okay, so a useful example can be cited with the help of the blood sample. So now you see large and more granular granulocyte cells produce a large population with high side scattering and forward scattering. So monocytes, as all of you know, monocytes are the large cells, and this is what is appearing here. This monocytes are having large sides, large forward scatter, which means the forward scatter is large. That is why it is appearing here. And for the cells of granulocytes, as the name indicates, they are having more granularity, and therefore the side scatter is more for granulocytes. Is it clear? So for the cells which are having more forward scatter will appear here. You can calculate, it is between 600 to 800. Cells having more forward scatter will appear here. Cells having more granularity will appear here. In between all the cells, if you take the lymphocytes or the lymphoblasts, which will be having which will produce like with less forward scatter and they are not so granular, so they have low SS, so which will be appearing here. So this is how the interpretation of the cell suspension will be happening based on the forward scatter and side scatter, and that will be converted into histograms. And compensation is yet another important term that you have to understand when you learn flow cytometry, which is required when analyzing more than one color, and we have to be very careful of the spectral overlap. And previously I was mentioning multiple fluorochromes can also be used, and in this particular site you can observe FITC and PE are used, and this overlap of this particular spectral overlap can be avoided by compensation. I think this slide would give you a better idea about compensation. You can see here X axis is labeled with FITC fluorochrome and the Y axis is labeled with PE fluorochrome. Okay, so now the segregation of the cells has happened, and you see this FITC is labeled here, and this particular cells are with the Y-axis, and this particular cells obviously has to be with FITC because there are only two fluorochromes. So one is with this PE. Obviously, the upper portion, the quadrants, the cells that are present in the third quadrant must bring down. This is what is called compensation. And now you can see two different population of cells segregated from a single cell suspension. Okay, so one is labeled with FITC. This is labeled with PE and this is labeled with FITC. So no need to confuse here. We have to choose the fluorochrome based on the cells. As I was describing, in a blood cell population, you will have multiple number of cells, say monocytes, lymphocytes, leukocytes, whatnot, and you have to tag one particular cell with monocytes means with FITC, lymphocytes means with PE. So like that, when you tag the fluorochrome with the particular cells, then after flowing through the flow chamber, you will be able to segregate the cells like this. So this is again also compensation done here. So one is tagged with FITC, the other one which is tagged with PE. Whatever here, most of the cells is already in the place, but still the final fine tune of compensation will bring the cells to the same lane, segregating two different populations. So now here again we can see the same after compensation and everything, the results will appear like this. Can you see the one present in this lower first quadrant is always cell debris. So we have to ignore the cell population. It may be debris, it may be dead cells or whatnot. So let us not bother about this quadrant always. So when you tag it with two fluorochromes FITC and PE, you will get two population of cells. This is one and this is the second one. So this is how the results of that will be appearing. So you can segregate cells into different populations. So from that, again, there is yet another possibility by gating. So what is gating? Here, we have taken this particular lymphocyte population, and within the lymphocyte population, subtypes will be available, and those subtypes can also be segregated by again combining to a specific marker. I think you all might have learned about immune cells and the markers present on the surface of those immune cells. So CD2 cluster differentiation, immunology, if you have learned, definitely you would have known about cluster differentiation. So this CD2 markers are present on the surface of one population of lymphocytes, and CD19 markers would be present in the yet another population that is lying in this particular thing. Okay, so what happens here, this lymphocyte population, after analyzing with the CD2 and CD19, again this particular after gating, this particular population, lymphocytic population, can further be segregated into two subtypes. One is the cells that are having CD19 markers on its surface, and another one is CD2 markers on its surface. Okay, so this is about how to interpret the results of flow cytometer. So coming to the applications, what are all the various applications that can be performed using this particular flow cytometer includes DNA content analysis. Okay, so this abnormal DNA content, sometimes called DNA content aneuploidy. which can be determined in a tumor cell population. So this DNA content aneuploidy helps us, this flow cytometer helps us to distinguish between the DNA that we are analyzing, whether it is a normal or abnormal one. If it is abnormal, then it is nothing but aneuploidy that leads to what is called determination of a tumor population. And the cell cycle analysis is the major breakthrough in flow cytometer. So any cell cycle analysis, how the cells are getting proliferated, especially in the cases of cancer diagnosis, we are very much particular whether we are very much bothered that the cell proliferation has to be arrested. So whether that is achieved by the particular drug with which we are interested or the phytoconstituent that we are interested. So we have to testify whether this could control the cell cycle operations or cellular proliferation. For analyzing that, this cell cycle analysis would be of great use. So in a normal cell, you can see the GO, G1. These are the phases of the cell cycle. So GO is the quiescent stage and this is the preparatory phase, and S is the synthetic phase of the cell cycle, and G2 again a preparatory phase, and M is the mitotic phase where the cell or the DNA replicates and divides. Okay, so what happens in a normal cell, we can find the GO-GN phase of the cell cycle would be of very high compared to synthetic and G2M, because it is a normal cell, only if it gets triggers, it will multiply. But when you go for a cancerous cell, the GO-G1 phase obviously get reduced. Instead, the S phase and the mitotic phase, because the cancerous cell is the immortal nature and also unlimited growth. It will be continuously proliferating with no entering into the quiescent or the GO phase. And therefore, you can obviously see for a particular cell, cancerous cell, this GO-G1 would be of reducing and the M phase and S phase will be increasing. But here, if you see, whether the cells are undergoing death, that can also be analyzed using flow cytometer, where you can find this particular S phase or the M phase is getting reduced because of this apoptotic cell death that is happening due to treatment with the phytoconstituent or the drug of your interest. And even chromosome analysis can be done. Erythrocyte analysis, that is fetal red cells in maternal blood, can also be analyzed. And this is the 4-color pattern, helps you to identify the immune cells having different markers, including CD56, CD3, CD56, if the marker is present, then it is a natural killer cell type, and CD3 marker is present means it is a T cell, and CD8 means it is a T cell, cytotoxic T cell, and CD4 means it is helper T cells. So all these cytotoxic cells can be differentiated by tagging it to a particular fluorochrome, thereby analyzing the surface markers of these cluster of differentiations in different immune cells. Reticulocyte analysis is also possible with the help of flow cytometry, and immunophenotyping of leukemias and lymphomas could be very well be achieved, and this particular is an essential aid for accurately diagnosing and prognosticating leukemia and lymphoma. This particular thing helps us to identify the phenotypes of B cell cancers. So based on the interpretations obtained here, we can categorize what are the markers that are present in different leukemia, like chronic lymphocytic leukemia, mantle cell lymphoma, follicular lymphoma, hairy cell lymphoma, marginal zone lymphoma. So based on this particular interpretation, we can arrive at a conclusion that CD19 markers are present in almost all types of B cell lymphomas, and CD5+ markers are present in chronic lymphocytic leukemia and mantle cell lymphoma, and this particular CD5 marker is lacking or absent in these type of leukemia. Like that, we can make interpretations, and also it helps us to identify. It helps in the identification of lineage specific antigens for perfect diagnosis. And immunological monitoring of HIV infected patients is also possible with the clinical flow cytometric instrument. Platelet-bound antibody analysis is possible. Reticulated platelet can be very well be quantified using flow cytometer, and the pathogenesis and molecular defects of many thrombocytopathies are identified using this flow cytometry. And these are the few published papers with the data incorporating the flow cytometry. So here we can identify the cells are segregated. Based on this, you can identify that what is the proliferation rate that is obtained after treating the breast cancer cells, whether the apoptosis has happened in the MCF7 breast cancer cells or not, can be analyzed using flow cytometer. The like the rate of apoptosis inducing ability of the sample to be tested, whether the whatever the drug or phytoconstant we are interested, we have to add that phytoconstant in the cell suspension and check for its apoptosis inducing ability. Only then, if the apoptosis is induced in a better manner, then we can declare the whatever the phytoconstant or the drug component that we have tested has very good efficacy against anti-cancer, especially for breast cancer, we can say with this particular picture. And so with this flow cytometry is done. So we'll move on to the next particular instrumentation is based on the electrophoretic method is two-dimensional gel electrophoresis. So all these techniques what I'm describing here is very much useful for analyzing the purity, quality and safety of the particular drug what you are developing. So after coming through all these high throughput techniques, finally you can arrive at a platform where your drug will be a right component of choice or what that can be decided based on the results of all these techniques that you are performing. So this, as the name indicates, it's a two-dimensional gel electrophoresis. So the first dimension is depend upon the separation of the protein molecules based on isoelectric focusing. So what is isoelectric focusing? Every protein will be having isoelectric point where at that particular pH, at its isoelectric point, the protein will acquire neutral charge. It will neither move towards anode nor move towards cathode. But when it is suspended in a pH gradient, when there is a change in the pH, obviously the protein will be separated based on its the IPH that is presented in the strip. So obviously the separation will be happening based on the charge that it possesses. So that is one dimension where you are separating the protein that is present in a mixture of proteins based on its charge. In the second dimension, again, in the second dimension, the same separated protein will be subjected to second dimension separation. Here, the principle or the separation is achieved by based on the molecular weight. If there is any change in the molecular weight, further the protein molecules will be separated. So that is why the single gel will be exposed to two different separation. The first dimension is based on isoelectric focusing, that is based on the charge it will be separated, and in the second dimension separation will be achieved based on the molecular weight. So now this is the schematic representation. We can see these are the two conditions, dimension 1 and dimension 2. Here this is based on the isoelectric focusing and this is based on the molecular weight. So now the protein molecules are separated and here this is again separate. Both the dimensions, the protein molecules are separated and which will be stained. And now you see when this is taken for detection or visualization of the spots and then detection through MS, you can identify like this. So these are the various steps. First, you have to prepare the sample, which is very important. In the next slide, I will be discussing on that. Then first dimension separation, second dimension separation, visualization of protein parts and identification. So the sample preparation, I said it is very, very important because you have to make the protein soluble in both the phases. Only then what happens? The first dimension as well as the second dimension separation of the proteins would be very great. The resolution would be better. Based on that, detection will also be very obvious. Okay. So in the first dimension, as I said, this immobilized pH gradient will be prepared, and because of that will be prepared using ampholytes. You must have heard about ampholytes. Ampholytes are the substances having both positive and negative charges. So using that variable pH ampholytes, we can prepare this immobilized pH gradient, often called IPG strips, and these IPG strips are allowed to be hydrated first and then it will be placed onto the apparatus and then current is applied. So now you see if there is an increasing pH, then obviously you can obtain the separation of the protein molecule based on the charges. The one which is having the negatively charged will be depositing on the anode, the other vice versa. Okay, then finally it will be going for equilibration. Later it will be detected using MS spectra or not. Okay, so now you see for the second dimension, based on the molecular weight, the separation would be achieved. So this is how the entire process will be happening, and the visualization. Now coming to the interpretation. So if you see the interpretation here, the proteins which are having similar pI, their isoelectric point will be similar to the two proteins that are having similar pI but with different molecular weight can be identified here. You can see these proteins are having similar pI, but they are having different molecular weight. So this is the advantage of this two-dimensional gel electrophoresis. If you are using only one dimension, both the proteins will appear as a single spot. Now we can see them as a two different spots because it is run in two dimensions. Here again, we can see these two are the proteins are having similar molecular weight but different pI. So what do we understand here? Because of that, if there is only one run based on the pI, you will get different spots. But if it is on the molecular weight, you will get only one spot. Because of two dimensions, we are arriving them as two different spots. So this is the protein identification mode. So these are the analyzers. So finally, after all this process, you can excise the spots and digest it and then take it to MS and MS spectra. So in the following slides, we will be discussing about mass spectra also. That time you will understand this better. So this is the database. So what are the 2D gel databases? It's there. It can be used as a reference library. It is available in the XPASI tool of the bioinformatics platform. So in the in silico, you can just refer with any of these library when you are working with an unknown protein. Okay. So these are the various applications of this 2D gel electrophoresis. Cell differentiation. This is particular whether the cell is getting differentiated or not. This is very, very important in particular with cancer diagnosis. So when you see the cancer diagnostic report, the biology of the tumor will be better understood by the presence of moderately differentiated cell, poorly differentiated cell, and very well differentiated cell. If it is a well differentiated cell, then we need not bother. The malignancy is poor. But if it is poorly differentiated cell, then we have to really bother. So in order to understand the biology of the tumor, this two-dimensional gel electrophoresis will be of helpful. Detection of disease markers, cancer research, all this can be of great applications related to two-dimensional gel electrophoresis. Next, so this is the paper that is published. So here again, you can see the cancer-specific protein markers that are obtained from the esophageal scan cell can be obtained by two-dimensional gel electrophoresis. And next, coming to the chromatographic methods. So HPTLC, I think most of you must have learned about the chromatography, paper chromatography, TLC, and so I am dealing only with the advanced level techniques, HPTLC, which is very popular because of visual chromatogram, multiple sample handling, and also enables the complicated separation. And I'm just skipping the principle because it is something what we've learned for the basics. So what is it is different, in what way it is different from the usual or the basic level of TLC? If you see, the efficiency is very high, the analytical time is greatly reduced, and also the solvent required is very less, and it is highly automated, and it is also commercially available. The spot or the bands will give you a very good interpretation to decide about the phytoconstituents that are present in the herbal extract or whatever the sample it is. So this is the instrumentation of the HPTLC, and these are the various steps that are involved in HPTLC, starting from the preparation of the plates, washing, sample application, mobile phase choosing, and the chromatograph development, detection and visualization. Later you can document it. These are the various steps, and these are the various applications. Once again, almost most of the techniques involves the applications in the drug discovery, food analysis, clinical, whatnot. Almost all these are the applications of HPTLC. So these are the results with which we can identify what are the phytoconstituents. So this particular paper cites about Annona muricata. This is helpful in identifying the phytoconstituents that are present in the extracts of Annona muricata. These many phytoconstituents are present. This can even be correlated with the help of the reference or the standard library that is available with any of the software that is coming with the system itself. And next to HPTLC comes the HPLC. All of us know it's a liquid chromatography, and again these are the different modes of HPLC separation. It can be of normal, reverse phase, adsorption, ion chromatography, or size. All these are having their own specific principles, and these are the components of the HPLC. And this is the schematic representation how the HPLC. So now for the HPLC, the sample is injected and it will be detected, and finally the HPLC profile will appear like this. So all these are the processes and everything, how the sample is injected. Instrumentation part, this is just an photography of this HPLC setup, and these are the HPLC, and this is the column. These are the columns that will be built inside the HPLC. So here, if you can see, the column will be the column box. It is given here. So this column will be present inside this box. Okay, so these are the columns. And what are the various detectors? I need not concentrate much here, because we know there are several detectors available. Depending upon the application or the nature of the sample that we are analyzing, we have to choose these detectors. And coming to the application, this is also having large number of applications, especially for the purification. Separation and purification of any particular compound can better be achieved using HPLC. And these are the few published papers where your HPLC profile will appear like this, indicating the presence of, this is again Annona muricata. You can find the presence of gallic acid, catechin, chlorogenic, caffeic acid. All these are present, appear as a peaks, major and minor peaks in the HPLC profile. Next comes the gas chromatography or the mass spectrometry. So this GCMS, it's a combined, the feature of this GCMS helps us to identify different samples within a particular test sample. So this is capable of separating, detecting, and partially characterizing the organic compounds, particularly when they are present in very small quantity. Okay, and this MS, the gas chromatography combined with MS, this MS provides some definite structural information from even a small quantity of sample. So that is what the idea of gas chromatography, that separate mixes of chemicals. So each can be identified individually, and a chemical analysis technique combining two instruments to provide for a powerful separation and identification capabilities. So combining these two will give you a very efficient result. So the sample solution is injected first, and then it is vaporized, swept onto a chromatographic column, right? And later on, this will be converted to ions, and this ions will be detected. So this particular ionic conversion is very, very important with respect to gas chromatography. So the analysis in the mass spectrophotometer forms a major, that is the ionization source, filtering and detection forms the major part of mass spectrophotometer analyzed after gas chromatography. So this is the typical GCMS system where you can find that is the sample is injected here, and this is the column I have shown through which the separation of the molecules will occur. And see, this is the ionization process where the whatever the proteins that are separated will be converted to ions, and those ions will be passing through the detectors, and which will be converted to an electronic signal. So this will derive a very good result, and we can even understand the structural features of the protein that would be separated in the sample of interest. And this is the image of the GCMS, and these are the injectors which will help us to inject the small volume of samples in the gas chromatography. And these are the columns of two kinds. One is the packed columns and another one is the capillary column. And these are the various detectors. Depending upon the nature of the sample to be analyzed, we have to go either for the non-selective electrodes or the selective electrodes. And this is about the ionization. I said it is very, very important aspect of the gas chromatography combined with mass spectrum. So now you see, if a particular, after separated through the GC column, if the particle has to be ionized through several methods that will be discussed later. So these non-ionized molecules will not be separated. However, the ionized molecules will be separated, and that will be achieved here in this aspect. Here the thermo spray ionization is happening. That is why the heated filament is kept here. This will be better understand in the following slides. So now you see there are different ionization modes: electron impact, chemical ionization, electrospray ionization, fast atom bombardment, and matrix assisted laser desorption. These are the various methods depending upon the nature of the sample that we are going to test. If it is relatively small volatile compound, or if it is a peptide protein non-volatile, then you have to go for electrospray. If it is carbohydrate, organometallics, peptides, or non-volatile, then you have to go for bombardment. If it is of nucleotides, then you have to go for what is called MALDI assisted ionization method. So after this, so what happens during the ionization process? So after getting separated from the GC column, it will appear as what is called a neutral molecule. So this neutral molecule will have to undergo ionization, and then fragmentation and ionization make them a charged molecule, before which it was a neutral molecule. After the process of ionization and fragmentation, it becomes a charged molecule. Now you see, once it is ionized, that is only the resonant ions are allowed to pass through the detector, and which will be detected. Help us to understand the structural features of the phytoconstituent. If it is going to be a non-resonant, it will be thrown away from the path. So this is the filter that we are using, quadruple mass ion filter. So now you see the entire system. So now the ion source is there, and it will be taken into ion selection chamber, collision cell, and fragment ion selection. So these are the various process means of which the ionization thing will be happening, and only the selected ion, fragment ions, will be taken to the detector. So that will be achieved by the filters like triple, quadruple mass spectrophotometer. So now you see what happens during ionization and fragmentation. This is here the example cited is pentane. So pentane is the one which will be having 5 carbon, CH2 present in it. Okay, isn't it? CH2 like that. You can find 5 carbon atoms, and each of the carbon atom is bonded with the hydrogen molecule. Now you see what happened during fragmentation. Due to the homolytic cleavage, you can find the formation of the fragments, and after this cleavage, both of them will acquire charge. So once it is acquiring charge, mass to charge ratio will be read, and that can very well be analyzed using the mass spectrum. So now you see the mass spectrum of the pentane. After the cleavage, you can see here 1 CH2, and here CH2, so 4 CH2 molecules are attached, making the mass to charge ratio 57. But now you see here the cleavage is taken in this way, making the mass charge ratio 15. So this is one type of cleavage has happened, leading to 2 fragmentation ion. This is the second cleavage. Here you can see 2 carbon atoms and 3 carbon atoms splitting, making it another 2 possibilities. So totally there are 4 fragmented ion present in the case of pentane. Pentane, which is having 5 carbon, there are 2 possibilities of cleavage, either cleavage here or the cleavage 2 and 3 atoms, and therefore there are 4 possibilities. So with these 4 possibilities, the mass spectrum would obtain like this. So with this, the structural features of any unknown compounds that you are going to analyze using gas chromatography combined with mass spectrum would be wonderful to analyze what are those structures present in those phytoconstituents. So this is how the GCMS or the peak fragmentation of that GCMS profile would be obtained, and this will be referred to the standard library with which we can understand the structural features or the what are the functional groups or the structural moiety of the phytoconstituents can very well be analyzed. So these are the applications of GCMS, and these are the published papers, and you could see the GCMS profile here, and these are the various compounds that we can understand or analyze after GCMS spectra. And next followed by LCMS. So the one difference between GCMS is LCMS is mass spectrum analysis remains the same. In the case of GCMS, it is the gas chromatography that separates the components, and in the case of LCMS, HPLC with an interface will be replacing the gas chromatography, thereby ensuring the high resolution of separation. So that is what here you can see the HPLC unit will be present, and again the quadruple filter and detector, everything remains the same. So this is again the most wide used column in the case of HPLC is fast LC column and micro LC column. This is again depending upon the nature of the choice of the sample that we are using. And this is how the sample will be prepared. I'm just running because we are arriving at the end of the session, so I have few more slides. So if you cannot understand during the question session, you can ask me, I will do it. So the sample preparation is very much concentrate here on the concentration. You have to concentrate the sample. It should not be a dilute sample, and also disturbance like sodium and potassium must be gone through desalting process through dialysis. Only then the pure sample will be very much helpful for us to analyze the structural compounds.
Yes, madam, we can proceed, ma'am. We can proceed.
Yeah, no, it's I think it's slow. I'm not getting the slides.
Okay.
So hope I'm not so very fast, sir. Participants
You
are
are
able to understand.
you are in the correct flow. You are in the correct flow only. You can proceed.
Okay, sir. Okay. So this interface I said, because we are using HPLC, we need to have an interface for connecting this particular thing. We need to have the ion source, ionized samples, and electrostatic lens, which are very much help to analyze the mass to charge ratio. So these interfaces are finding very important in the case of LCMS to make the resolution very fine and also the structural details in elaborate. So these are the various interfaces that are used after HPLC before ionization targeting. So electrospray ionization, atmospheric pressure chemical ionization, and thermospray ionization. So these are the pictorial representations where this ionization process is happening. So we can see here a nebulizer is just pouring, so thereby it is getting converted, and all that is getting deposited in the electrode, and that will be analyzed here. Okay, so same way, if you see in the previous case I was showing, if it is a thermospray ionization, then you will have a heater instead of any vacuum or what is that, in the previous we have seen, no, instead of having the capillary tube or the nebulizer, you will be having what is called this heater to convert the ions from the separated the samples that are separated from the HPLC, and that will be taken to the detector. Okay, and this is the mass analyzer. So for this, you will always previous also I've told the MS quadruple mass filters are used. Time of flight is used, and ion trap is also used. Apart from that, you have what is called Fourier transform, ion cyclotron resonance. All these are the one help us to detect the mass spectra. Okay, so this quadruple mass analyzer is help us to detect in a very efficient manner compared to the previous methods. So the time of flight, this is nothing but with without the use of any electric or magnetic field, it is possible for us to separate the ions based on the time. That is why it is called time of flight. And also here, in the case of ion trap mass analyzer, this makes use of an electric field. Thereby, the separation of the ions are achieved, and that leads to mass to charge ratio, which will be efficiently detected by the detector. And then this is the FTICR. Here again, that is this uses instead of electric field, magnetic field will be used in order to trap ions into an orbit inside that. Okay, and in this analyzer, there is no separation that occurs. Rather, all the ions of a particular range are trapped inside, and an applied external electric field helps to generate a signal. So these are the various applications of LCMS. These are the few published papers with the results of the LCMS, and we can see these are the various compounds carrying the particular structure, and the functional group can very well be analyzed using LCMS. And coming to the confocal microscopy, this is an advanced imaging technique that is used for increasing the optical resolution as well as the contrast of the micrograph. Okay, so these are the instrumentation part. I'm just skipping it. This is the principle. So the important thing is, based on these two ideas, this resolution of this particular imaging technique is very good. Point by point illumination of the specimen, rejection of out of focus of the light. These two are the important aspects. Covering this, it could give us a very good photograph. So the confocal microscopy allows analysis of the fluorescent labeled thick specimens without physical sectioning. You can analyze the internal architecture of an organism. So that is feasible because of those two parameters. And this is the 3D projection. And more color possibilities are also feasible in the confocal microscopy. So because of that, the images are detected by a computer rather than by eye because of more color incorporation. So it is possible to detect more color difference, thereby the architectures, internal architecture of a cell or organism will be better visualized. And these are the confocal microscopic images, which will give a better see for this is the kidney cell. So fluorescence versus confocal microscopy. You can see in the fluorescent microscope, though you are tagging it with a fluorochrome, it will be appearing like this. Whereas in the confocal microscopy, the architecture of the cells, including the nucleolus, nucleolus, everything will be very prominent, and the matrix region, everything will be very prominent in the case of confocal microscopy. So this is again then a confocal microscopic image, and this is the published. So here again, like the presence of the cancerous cells, ignoring the normal cell presence, can be observed readily in the case of confocal imaging. So when you test the sample, you can process even the blood sample and then stain it appropriately with the proper stain, maybe cyber green or Hodges stain. You will get a better illumination of the cancerous cells rather normal cells. So it will help to confirm the presence of cancerous cells in the sample.
Yes, madam. What's happened? Proceed. I'm bringing it down.
Hello.
Yes,
Sir,
madam. Yes, you can proceed. Proceed. Yeah.
what happened? My laptop with no charge, I couldn't run my slideshow. I'll just
Okay.
coming in another line, sir.
Yes, yes, madam. You can proceed. Yeah, we'll wait. We'll wait. No problem.
You're sorry for the delay.
No, no, no problem. No problem. Proceed, madam. I think due to a technical issue, madam will rejoin within 5 to 10 minutes.
Yeah, the size of my PPT is very big. That is why, sir.
Okay, okay, no problem.
Is there any doubt? I think I can address. If the participants are having any doubt, I think we can discuss now.
Open the session.
Now, I think it's okay, madam, so that you can go over the concluding the session also, if you're, or if you want to continue also, you can.
No,
continue.
I have only a few more like arrays, microarrays, protein microarrays, and DNA microarrays only I need to discuss. All other techniques I have discussed. Whatever the techniques so far I have.
Yes, madam, we can wait, madam. We'll wait, no problem. They're ready to wait also. Audience also ready to wait, actually, no problem.
Sorry.
Okay, okay.
Okay, okay.
Because they are much interested knowing about all the techniques. So we request all the audience to just wait for 5 more minutes, because madam is having some technical issue, and he may rejoin within 5 minutes, right? So if you have any queries or any questions, you can post it in the chat box. So we request all the participants, those who have any queries, you can post it in the chat box. Those who are watching live in the YouTube, we request all the participants to post in that of your YouTube chat box also. Those who are in the Zoom platform, you can post in that of your Zoom chat box. And those who are in that of your YouTube, you can post in the YouTube chat box. So we request all the participants to wait for 5 more minutes. And Madam is having a few slides for, I think, related to array. He may be having some slides. So we may be rejoin within few minutes. Please, we request you all the participants to be patient and wait. Thank you. Thank you so much.
There is no query from the participants. Any doubt also I am ready to clarify.
Madam, there was one query that whether some interesting queries there.
Yes.
So, yeah, is there any course for the master that advanced in techniques? He is asking. Somebody.
No, sir. Sir must be Dr. Karthikeyan must be knowing. I am in charge of advanced research laboratory.
Yes. You can,
Very
you
frequently.
can tell. Yeah, you can tell about that your facilities, and you can collaborate. Those who are interested to do any of the work, you can collaborate with Madam. I think so. Madam will be telling about that. Actually, you can proceed,
Yes,
Madam.
sir. As actually we are having that advanced research laboratory. It is actually the main objective of this laboratory is to outsource samples. So most of the techniques what I have shown are we are outsourcing such samples, especially flow cytometer, HPLC, and all animal tissue culture techniques we are doing. And if you give your sample, we will analyze and give you the results. Even interpretation, we are ready to help. And regarding the course, we don't have course as such, but very frequently we conduct workshop. Hands-on training would be organized, during which time it will be thrown out for the participants from other institutions, and they will participate, and they'll get, maybe it is for 2 days or 3 days program we will do. And during which time you can register yourself for the workshop, and then you can have hands-on training. And sometimes we will give you opportunity to analyze your samples if you have 1 or 2 with you. If it is 2-dimensional gel electrophoresis workshop, then you can separate your protein samples. You can bring your sample, and then after attending the workshop, you can analyze and get the results in hand and go. So this is what the workshop very frequently we organize. So those who are interested in joining the course, rather sir, I think mine is
Yes,
any
madam.
other question?
No, madam. Actually, they are very much interested. Actually, and,
it's not a problem if the session goes beyond 12:30.
no problem, madam. You can proceed.
Okay, sir. Okay, shall I proceed? Okay,
Yeah, you can proceed, madam. You can proceed. Actually, no problem.
Okay, sir. Okay, sir. I'm coming online.
Okay. Thank you so much. So all the participants, we request for wait for few minutes, and I think Madam will be rejoining within few minutes due to some technical issue. Madam will be rejoining now. So we request all the participants to wait for few minutes.
Actually, I forgot to incorporate, put it in the charge, sir. The charge went off, so the presentation I could not display. That's the problem.
Okay, okay. Now it's okay, or madam, or
No, I've connected. The charge is connected to my system. I
okay.
think it will be working in another 1 or 2 minutes.
Okay, okay. We are ready to wait, madam. No problem. You can proceed.
Desktop ல தான் இருக்கு. போயிடுச்சு.
Hello, madam. Hello, ma'am.
Sir, I'm coming.
Yeah, yeah. Okay.
Sir, some of the questions they are asking, maybe due to COVID-19 pandemic situation, right now we don't have plan of organizing any workshops. Maybe it will be executed in the following days, maybe in the next month. Sir must be knowing, Dr. Karthikeyan must be knowing, we got lots of trouble in establishing the animal cell culture. So we've lost all the cell lines because of that COVID-19 pandemic lockdown. So
Yes,
maybe next month we may plan, so I can communicate with Dr. Karthikeyan, and he may let us, the participants, know about the workshop that we
Sure,
will be
sure.
conducting. And the another
Sure,
question:
sure.
do you provide NMR spectroscopy and its interpretation for natural compound? Right now we don't
Yes.
have NMR spectroscopy. We have only FTIR, and we have a very good laboratory called CNR Rao laboratory, where scanning electron microscopy is very well done, and for which very frequently we organize a workshop. And that is also, I'm just giving the link. It is available in our website. So the website is www.avinity.ac.in. So if you get into the Avinash Lingam University website, all the details of this workshop will be displayed, especially scanning electron microscope run by CNR Rao laboratory. And I am in charge of advanced research laboratory. And this particular scanning electron microscopy and FTIR and spectral analysis, X-ray diffraction analysis, all these are available with us. And that is all the information is available in our university website, Avinash Lingam Institute for Home Science and Higher Education. Any other questions? So many are telling informative. I felt it was very speed because I had to discuss too many techniques with lots of principal instrumentations and methodology. I felt it was very speed. I ran it in a very speed manner, but I'm very happy that all of you could understand what I was dealing so far.
Yes, madam. Actually, nobody is told about any speed or you are going speed. Nothing. No comment
Okay, I
related to
was
that,
running
because
my
all
slides
are
very
all are satisfactory, and they are very much
fast.
interested also.
Okay, so only this interruption is making
No, madam. No, madam.
technical
Proceed. That
issues.
is online now. We can know that.
Because of my PowerPoint is very, like, it was 25 MB, sir, size,
Okay.
because I confused many images. Because of that, I got this technical issue.
No, madam, no problem. There is another question we can see that, madam, is any analytical techniques?
Analytical workshop is essential. There are two workshops we organize. The one is the basic techniques in biological sciences, and the second advanced workshop is advanced techniques in biological sciences. So if for the fresher, I would suggest basic techniques in biological sciences, where we will deal with how to analyze a sample fresh. That is again not with the HPTLC, and for chromatography means it will not be HPTLC. Instead, we will be showing you how a compound can be separated using TLC, column chromatography. So in the advanced instrumentation like HPLC and HPTLC and GCMS and all, I've shown you the prepared column in the photographs, images. Hope you remember. But in the case of basic technique, where we teach how to prepare a slurry and how to layer it in the slides, and how to spot the sample and get the separation. So in the simple thin layer chromatography itself, we will get the spots and which will be visualized. And for fresher, we will teach such basic techniques like TLC, paper chromatography, and serum separation, one-dimensional gel electrophoresis, and all that will be there. Once you complete, then you can register for the advanced techniques, where starting with animal tissue culture, we will teach everything, how to use a flow cytometer, and everything will be taught in that advanced. Two-dimensional gel electrophoresis will be taught, and many things will be discussed in the advanced electrophoresis workshop.
Thank you, madam. Thank you so much.
Okay, sir. Bye. And the laptop connect பண்ணலாமா? Give feedback kudu. ஏன் போக மாட்டேங்குது? சரி, அந்த Sir, it is telling the meeting is scheduled for 29/8 and it will be started at 9:30. So it is not allowing me to enter now.
What, madam? It is not 29th, madam. This 30th, na?
But it is saying that you can join only on this day, like that. It is telling now. Because
No, madam.
morning
Just, no, madam. You don't think, madam. Just you go to the Zoom and
Okay.
your account, and just join. You click that as join. In that, you give the meeting ID and password.
That's what I'm doing it, sir. But still, it is saying when I say join meeting, it is giving
Today's meeting ID, madam.
today's meeting ID. 29th கொடுத்திருக்க.
Send both IDs for that 29th and
No,
30th.
sir, both ID is same, no?
Different, madam. Both is different.
87922.
822-9060-2526.
No, madam. That is different. That is yesterday's actually.
Okay.
Today's is 879-233-5975. Like that. Yeah.
So meeting passport, please tell me, sir.
Password is 289083.
So I gave the yesterday's
Yesterday's,
meeting.
yes, yeah.
That's why I could not join.
Yes. Yes, madam. We are joining.
I'm
now.
getting connected, sir.
Yeah. Fine. I think you are joined. I think
Sorry
so.
for the interruption.
No. You can proceed. Madam, you can share your slide actually.
Yeah, I am sharing it.
Yes, madam, we can proceed actually.
Okay.
Yes.
So we stopped here. Protein microarray. It is yet another important and most significant experiment with respect to diagnosis of cancer, especially because we all know cancer is comprises of more mainly due to cancer occurs mainly due to mutation, and there is a change in the genetic profile. So that can very well be analyzed either making use of protein microarray or cDNA or any other thing. So whatever the genetic changes that are happening, obviously it will be reflected in the synthesis of protein, and that can be very well be analyzed using protein microarray. So this protein microarray, as I said, it is a high throughput screening method used to track the interactions and activities of the proteins in order to determine their functions, and which will be due to mutation or some other lifestyle related disorders, the functioning of the protein may be changed, which will be reflecting as the consequences may lead to cancer or any other degenerative disorders. So this protein microarray are rapid, automated, and highly sensitive. Only small quantities of samples are very much required to do this experiment using protein array. So this is the schematic representation where this will be depending upon the preparation of the protein chip. So in this, either the antibody, protein, or peptides, all are protein in nature. As everybody knows, whether it is an antigen or antibody, it is of protein in nature, which will be fixed onto the slide, and then it will be incubated with the biological sample with which you are interested in analyzing. After incubation, you could see if there is any complementary feature that is associated with the sample and the antibody or the protein of interest, which will be binding onto this, and later this can very well be detected using any of the, after scanning, if it is tagged with any of the fluorochromes, this can be very well analyzed as a signal, electronic signal, and that data can be analyzed after transferring it to a software, and the heat map will be generated, will make us better understand the differentiation profile of the protein that is getting expressed from the genetic makeup of any individual organism. So now this particular schematic representation will be very clear when we move on to the further slides. So this these are the various steps in protein microarray. You can find this protein chip consists of the support surface on which it may be a glass slide or a nitrocellulose membrane or any microtiter plate. 96 well plate, some of you must have seen, and that will serve as a protein chip where you have to allow the proteins to bound onto the substrate. That is the fixed solid support where you have to allow the protein samples to get fixed. And then after fixing, the probe molecules that is typically labeled, as I said, it has to be labeled with fluorochromes, only then later you can stain and detect as an electronic signal. For to enable this, you have to allow the labeling of this fluorochromes to this probe molecules that is bound onto the solid support. And then now the array is ready, and now you have to add the sample to the array. If the sample has any complementary association with the probe that you have prepared, then the both of them will bind, and you can separate the complementary probe which is having the association from your sample can very well be identified. The rest of the one which is not having any association with this protein molecule will be washed away, and the one with for data analysis. Okay, so these are the two different types of arrays. It can be analytical protein array or functional protein microarray. And this analytical microarray, next slide will be describing in elaborate. And this functional protein array where the binding activities, that is protein interaction, protein DNA interaction, protein lipid interaction, or protein peptide interaction is enabled in the case of functional protein microarray. So now I said this analytical protein microarray, this is how it will be. So there are two more. One is the sandwich mode, another one is antigen capture mode. This is the solid support or the inert matrix support we are using, in which the antibody is fixed. Okay, so now to the fixed antibody, this is the protein of interest. We are working on, we want to analyze this particular protein sample using the protein array. So this is the sample, and the sample is getting bound to the antibody that is impregnated with this inert support. And now, if it is a sandwich method, this protein sample will be again allowed to bind to the secondary antibody, and this secondary antibody in turn is associated to the fluorochrome, which enables us to get the data at the end of the detection. Okay, whereas in the case of antigen capture, this secondary antibody is missing here. Here the fluorochrome is directly attached to the protein of interest. So that these are the two different methods. Whatever the mode it is, you are having the inert support onto which you either the antigen or antibody is attached onto the support, and the protein of this in particular probe is present or not will be checked at the end of the experiment. And always this particular thing is attached to the fluorochrome, and it is the functional protein microarray where I said it is the protein-protein interaction, protein-lipid interaction, protein-DNA interaction, all that can be very well analyzed using the functional protein microarray. And this is the reverse protein where you can see the instead of the capture antibody, here the antigen is fixed onto the inert support, and you are allowing the antibody to go and bind to the probe. So this is the forward where antibody will be captured onto the support and antigen will go and bind. And in the reverse phase, antigen will be fixed onto the support and antibody will go and bind. So this is again the difference between what are the fluorochromes that can be used. The attachment of tagging of the fluorochromes to the antigen or the antibody will help us to get the data at the end of the experiment. So it can be fluorescent dyes like cybergreen, or the enzymes like harsh radish peroxidase, which will produce a color which can again spectrophotometrically measure, quantify, and the radioisotopes such as phosphorus 32, 33, carbon 14 can also be used. If we do not want to rely our experiment based on any of these fluorochromes, you have alternative methods are also available, something like plasmon resonance spectroscopy or optical ellipsometry, something like that. So coming to the applications of this protein array for diagnostic purposes, especially cancer, new disease markers can be identified, and protein expression profiling, that is whether there is in the previously I was telling any cancer cell, when you see the report, it will be given either as a poorly differentiated cell or a differentiated cell. If it is a well differentiated cell, then we need not bother about the malignancy. Malignancy is very, very poor. If it is a moderately differentiated or poorly differentiated cell, means there is malignancy present. So such kind of expression profiling can be featured using this protein array. Protein functional analysis can be done. So this is the functional protein microarray, and antibody characterization can be done. So plasma therapy for coronavirus, we are telling for the specific coronavirus, the antibody would have been generated, and those antibody would have been taken from the bed samples of the cured patients, and that can be used. So that is plasma therapy. So such characterization is feasible using this protein array. And the treatment, that is prognosis and diagnosis and prognosis for the several neurodegenerative diseases are also possible with the help of protein microarray. So these are the few published papers carrying the microarray analysis of the human non-small small cell lung cancer. So you can see here, all these are the patient sample. So there are 17 wild type samples present here, and there are 8 mutated samples are there, which means they are the sample of these patients are they are cancer patients. Okay, so now when you go for protein profiling, you can, this will be developed into 2 clusters, one minor cluster and one major cluster. This alone forms the minor cluster, that is the patient sample. They are all mutated sample and they are like carcinoma. And here you can see the major cluster, all are wild type, non-cancerous one. So this type of interpretation would be obtain, you go for protein profiling, and thereby you can identify the particular sample that you are analyzing, whether it is cancerous or non-cancerous. Here again, the x-axis is based on some of the protein tyrosine kinase markers, and in the y-axis again some other protein markers would have been tagged, and therefore the clustering will help us to enable these are the non-cancerous ones and these are the cancerous ones. Like that, the cluster analysis will be possible with the help of protein array. Next comes DNA array. So as I said, if there is any change in the genetic makeup, that will be reflected in the protein expression. Same way, at DNA level also, it is possible for us to analyze the array. So here, instead of protein, here we will be using the DNA, either the DNA or RNA. If it is RNA, it will be converted to a cDNA with the help of reverse transcriptase enzyme. So DNA chip, gene chip, DNA arrays, gene arrays, and biochips are the synonyms, where to the inert support, the DNA molecule will be acting as a probe, and to which you will allow the sample molecules to complementary bind, and thereby we can interpret the results at the end of the experiment. So this is the DNA chip technology, where you can find here, this is the DNA chip, which will be manufactured to contain hundreds or thousands of synthetic single strand DNA sequence. So now you see the unknown DNA from a patient will be separated into single strands and enzymatically cut and labeled with the fluorescent dye, and this will be allowed to fall on this. And if this has any complementary with the existing probe in the DNA chip, our samples, some of the samples are having the complementary portion that is binding. So after binding, when you wash it, the undesired or the unwanted or the one which is not bound to any of the sample component will be washed away. The one will bound will be forming. So this is the data interpretation, where you can find the tagged DNA will bind only to the complementary DNA on the chip. The bound DNA will be detected by its fluorescent dye and analyzed by a computer. The red light seen here is a gene expressed in normal cell, and green is a mutated gene expression, and yellow color represents both. Okay, so this is how we can understand, we can distinguish a normal cell and a cancerous cell. In normal cell, you could see some of the genes that are expressing and some of the genes are not expressing. But obviously, in a cancerous cell, you can see some of the genes which is not expressed in a normal cell would be expressing in a cancerous cell. So this kind of heat map will allow us to differentiate between a normal cell and cancer cell. Thereby, it helps us in diagnosing at the genetic or the DNA level. So this is what is the DNA microarray, which requires a DNA chip, a target sample. This may be a fluorescent label, only then at the end it will be helpful for us to analyze, and the enzymes required, fluorescent dyes, probes, and scanner. So this is the design of the array. So I was telling, this is your sample. You have to just purify it and make it mRNA using the enzyme reverse transcriptase. So this every mRNA can be converted to a complementary DNA with the help of the reverse transcriptase enzyme. And this cDNA is coupled to the any fluorescent dyes. Here the cyber green dyes are given. So this dyes, when it is tagged to the cDNA, then it is allowed to hybridize with the DNA chip, and after hybridization is done, you will be allowing taking the slides for washing. After washing, you will get only the samples which got the complementary portion to bind to the probe alone will be retained, and that will be observed. Finally, the data will be analyzed. Okay, so this is how to prepare a DNA chip using photolithography. This is not required because the DNA chip is commercially available. We can purchase that and use it. So these are the two types of DNA chips, or we can say microarrays. It is cDNA based or oligonucleotide based. Okay, so the cDNA based, here the cDNA I showed in the previous slide, from mRNA using reverse transcriptase cDNA will be prepared. And this is the oligonucleotide base, where the DNA chip with attached oligonucleotide sequences. These are the two types of chips. And this is the comparative microarray hybridization. You can see cell population you will be taking, isolation of extraction of mRNA, cDNA preparation, labeling it with the fluorochromes, hybridization to a DNA microarray chip, and then you are taking it for scanning. This is a detector. The laser beam will be focused onto the hybridized thing, which will be detected, and this is how the scanned image. The previous heat map I've shown. So that is how this is how it will be looking, and then from this you can distinguish between a normal cell and a diseased cell. So these are the various applications of the DNA microarray, and this is one of the published paper including the gene expression profiling of a breast cancer cell using the microarray approach. So now you see this is the heat map generated, and you know the breast cancer cells are of different kind. Either it is estrogen receptor positive or estrogen receptor negative. Sometimes HER2 positive. What is estrogen receptor positive? If it is having the binding capacity to the estrogen receptor, those breast cancer types are classified as estrogen receptor positive. And if it is having a HER2 protein present in it, then it is HER2 positive. Like that, different types of breast cancers are there. So this is one particular example where you can differentiate among the cancer patients those who are estrogen receptor positive and estrogen receptor negative. So such kind of findings are feasible with the DNA microarrays. So this that is what is meant by differential expression. Differential expression is one particular protein will be upregulated in a cell, and the same protein will be downregulated in some other cell. So that is what is meant by differential expression. Also, we can say a particular protein would be expressed in a normal cell, which will not be expressing in a cancerous cell. Same way, particular protein will be absent in a normal cell, and that will be upregulated in a cancerous cell. So such kind of differential expression can very well be analyzed using this protein microarray or DNA microarray. And the hierarchical clustering I've shown in the previous slide, which helps us to analyze. There are 278 and 334 differentially expressed genes after genistein, and here the sample they are taken for analysis includes a pure compound genistein and diapsine, and these two samples were were used for treating the MCF7 and MB231 and MCF. All these three are the breast cancer cell lines. Okay, so this is a non-cancerous breast cancer, non-cancerous breast cell line, and this is a breast cancer cell line, and this is again estrogen receptor positive cell line. These three are the three different cell lines, and the results are shown here, where the treatment of all these breast cancer cell lines with genistein and diapsine shown that there are so many different genes are differentially expressed. That is what is analyzed from the hierarchical cluster that we are deriving at the end of the microarray experiment. So what is 72H? The experiment is that is the treatment of these two molecules to the cell line was happened for 72 hours. Okay, so the next, the final technique that we are going to discuss in today's session is RT-PCR, that is reverse transcription polymerase. Why I have taken finally included? Because due to COVID-19 pandemic, all of us are aware that the coronavirus can very well be diagnosed using this RT-PCR technique only. So we need not get confused with the qPCR and the RT-PCR. In the qPCR, like, it is just help us to separate and distinct technique, whereas RT-PCR is the one which is used to quantitatively measure the amplification of DNA using fluorescent probes. Okay, so this is the schematic representation of the technique where RNA is present and you have to use the suitable primer to amplify it. After amplification, with the help of the enzyme reverse transcriptase, this will be converted to cDNA and it will be taken for polymerase chain reaction to get the amplified DNA, and those amplicons are finally assessed for their presence of the particular viral DNA is present or not will be assessed from the amplicons of the PCR experiment. So let us not get confused between the PCR and the RT-PCR. In the normal polymerase chain reaction, the three essential steps involved, which includes denaturation of a particular DNA. Once the denaturation is happening, it will lead to the formation of the single strand. Only then the you can allow the combination of primer with that, so that the synthetic process will be taken in after annealing process, and finally it will be extended and the DNA molecules will be amplified. So in the case of polymerase chain reaction, the amplification of the DNA will be happening. And what is RT-PCR is, you have to take the sample, I extract RNA from the sample of interest. You know, if anybody wants to test whether we are infected with coronavirus, we have to take our blood sample, isolate RNA from the blood sample, use the respective or the appropriate primer for the coronavirus, and then take it for go for reverse transcription. cDNA of that will be produced, and using this PCR technique, you can amplify and get the amplicons ready. Once this is done, this can be checked with the library whether it is giving positive result with coronavirus or not. Okay, so this RT-PCR can be achieved in two methods. One is the one-step procedure, and the second one is the two-step procedure. In the one-step procedure, directly you will add the appropriate primer and do the RT-PCR. And in the two-step procedure, it is like not specific. In the one-step procedure, you are using a specific primer, and in the two-step procedure, you are using a non-specific primer, then reverse transcriptase. Finally, you are getting the amplicons here in the two-step RT-PCR, right? And these are the difference between the first one-step and two-step procedure. Let us not go. So this is the application of the RT-PCR, where for most of the research methods use this either qPCR or RT-PCR. Gene insertion is possible. Genetic disease diagnosis is possible. Cancer detection is also possible with the help of RT-PCR. So now this is a research article published like a real-time PCR assay for bad SARS-like coronavirus detection. And you see, out of the samples analyzed, only this sample is giving a positive result with the coronavirus. So that not corona, SARS-like coronavirus. So you can see why so many bands are appearing. This A, B, C, D are the varying concentrations of the sample, even at lesser concentration. And W is the without template. Without the particular specific template or the primer, if you do, there will not be any amplification. So it is run without the template. If you put, there is no formation of the amplicons. In the presence of appropriate primer, you can find the presence of SARS-like coronavirus. So by decreasing the concentration, you can see, obviously observe the change in the intensity of the band octet indicates the concentration has reduced. And this is again yet another test to diagnose the coronavirus. So now you see, this is the plate where you can find this is control and this is COVID-19 and this is influenza. So the results told it is a negative result. Only this band is appeared, and neither this nor this appeared, and therefore this particular is a negative result for the diagnosis of SARS-like virus. Thank you.
Yes, ma'am. Thank you so much. And I think the most of the queries you have just been cleared actually during the technical problem. I think so.
Yes.
Actually, I would like to conclude the session because session has gone somewhat time extended. So I'm very much thankful to you, madam. Actually, it was new experience for me also. Even though there's slides you are experiencing, there is more than 170 slides are there. But you are just with the full energy from starting to end. You have the full energetically you made as to be watch attentive, and so that is I'm very thankful to you. I think most of the students, it will be very useful for them for doing their research work, or the PhD scholars and the PG scholars, those who are doing the research. So it will be very useful for them because madam has advanced technique. He is explaining the basic principle and their applications, everything starting from the flow cytometry, HPLC, HPLC, and what are the different array metrics as well as electrophoresis, all the things he elaborately he explained. And it will be very useful for the top your UG, PG, and PhD students for doing their research work. So thank you so much, madam. And on behalf of Sydus Publishing and Kalinga and Biomedical Research Institute, I am Odisha. I'm very much thankful for you. And upon invitation, you accepted the invitation, and you're ready to give a talk. I'm very thankful for you, man. Thank you so much, madam. Thank you.
Thank you, sir. Thank you so much, sir. Thank you.
Okay, thank you, thank you.
Thank you, ma'am, for your energetic and informative session.
Okay, thank you. But sorry for the
Thank you.
interruption.
No, madam, that is
No,
normally
no,
happens. No,
it's
thank
online.
you.
Okay, sir.
Okay,
Thank you all. Thank you all the participants.
Thank you.
Thank you.
Yes, yes, thank you.
Okay.
I welcome all the participants to validatory function.
Yeah, can you please? Yeah, please. Yeah, please. Thank you. Yeah.
On behalf of Sydus Publishing India, I would like to invite Dr. Subandhu Mishra, Coordinator, Director, Kalinga Biomedical Research and Establishment, Odisha, and Dr. Karthikeyan, convener of the event, to the validatory function. I would like to request Dr. Subandhu Mishra to give the speech, guest speech. So please.
Good afternoon, everybody, and good afternoon, Karthikeyan. And
Yes, sir.
nice session. I think I'm got missed on the last day, but this is a nice session, and whatever the innovative things which is I learned. So before I did the validatory functions, I just like to introduce myself and my research institute, whatever the research work is going on in the institute. I like to I'm Dr. Subandhu Mishra, having 10 years experience, and I have started Kalinga Biomedical Research Institute in before last 2 years, and I'm just showing a slide on the our research appearance. It will be. Can I? So that is Kalinga Biomedical Research Institute. It is the contract research and professional training organizations that should be used of the field of pharmaceutical science and life science, and it will be offer the high quality bioanalytical service like undergraduate, postgraduate, and the PhD scholars, and also the sample testing analysis of different area, like we have having HPLC, we have having UV, we have having IR. So different testing instruments we have, and the testing materials we have also testing to the also the training program to the different undergraduate or postgraduate. And also we have the different laboratories like microbiology, pharmaceutics, pharmacology, and we have the different important part of the practical knowledge to the students of B.Pharm, BSc and M.Pharm student for the dissertation work. In our also the experience and the efficiency of the faculty members are increased to the different competitive, and also the guidance of the different education program and social economic careers and other things we have posted to the different organizations, and also collaborated with a different institute. So our collaborations are basically in the different areas, that is we our consultant like Mr. K.K. Patel, the DRA head of the Z laboratory, and Asit Kumar Sahu, that is the associate professor of Silsa College of Pharmacy, Dr. A.K. Singhai, the director of NLOCIPBS, and also the Mr. Pramit Roy, that is the team leader of Trials, and Dr. Sachin Dubey, scientific collaborator to the Denmark, and also Mr. Prabhajan, the director of Scan Research Laboratory Bhopal. It is a collaborated of the different organization to guidelines to help to the process the research work in the future prospect. And our small teams, that will be I am the directors of this institute. The Mr. Nipun Bhoi is the research officer of the M.Pharm Pharmaceutics. The Mr. Supratik Roy is also the research officer. Ms. Shivani Verma, the research officer, MSc in Biotechnology. And we have concluded that is the training and the research work on our laboratory. Now we have the different departments, also the instrumental analysis, biotechnology, microbiology, chemistry and natural products, and also the department of pharmaceutics. So in our service, we have basically the outsourcing the service, that is the analytical service like HPLC, method development, UV spectrophotometer, zeta potential, NMR, FTIR, mass spectro and scanning electron microscopy. We have provide some instruments like scanning electron, mass, FTIR. We have got in the outsourcing, and but in our lab that will be HPLC, UV, particle size analyzer. We have provided to the samples. If you are need some research, you need some the result of the sample, you can send all these services, analytical services we have provided. Next we have the pharmaceutics and industrial pharmacy. We have research on the nanoparticles, liposome, phytosome, invasomes, all the training session of the tablet, capsule, syrups and other and also the quality assurance program, and also the stability studies and preparations we have in our lab. And also the microbiology biotechnology department, that will be microbial technique, that will be immunological diagnostic, water analysis, rDNA, immunological techniques, gel hydrophoresis and others program that will be shown in the slides. We have like the training session, that will be microbiology and biotechnology. We have trained to the some students those who are interested for the industrial purpose. So these we have internship program also for these techniques we have used. The pharmaceutical chemistry, we have isolations parameter in the research cells. We have different isolations techniques are there, that will be crude draw characterization, isolation, structural illustration and extraction, phytochemical screening, and also the chromatographic study like TLC column and HPTLC. We have research on this lab. And also other service like it will be provide the different students to the environment, to the dissertation work, and also the student who has prepared for the research papers. We have helped to the students which mainly use the area, and also we have used to the M.Pharm student for the dissertation work, and also MSc students, PhD, and guide for the curricular and co-curricular developments, and arrange to the career development program like innovative lectures, seminars and workshop of international deputies. We have the consultant, we are those we are related to the other students. So who are the if any type of need in consultation, you will contact our lab, and that is in the Dairipalli Phatak, Budaraja, Sambalpur, Odisha, where the contact number is there and the mail ID is there. So I just show the different galleries, different type of pictures which is as a lab, that will be UV and HPLC, that is IR, the HPLC that will be there, and we have the dominant faculty, that the development program is going on, that will be, and also the different instruments work, and that is the different students we have appearance for the training sessions. That's enough presentation. So thank you. If any query about this thing, then send me mail ID, and if any type of requirement, if you are for the research activity and any training program, you can call me in any time. Thank you. Over to you, Karthikeyan.
Yes, yes. Thank you so much, Dr. Subandhi Mishra sir, for your introduction about your lab and facilities. And I think it will be useful for them, those who are doing UG, PG and PhD research. You can contact myself, or I can provide you the address. After the session, I am ready to share that of your address and everything related to that of your Subandhi Mishra sir's lab. So it will be useful for you, those who are doing the UG, PG and PhD research, or the faculties, those who want to do any kind of research, you can contact me. Thank you so much, sir. We'll move to the next one. Yeah, we'll move to
This is the most interesting part of the validative function. That is the best poster award. I would like to request Dr. Karthikeyan, convener, to declare the best poster award. So please.
So as we know that actually today, the most of the student researchers, faculties, and also research scholars, those who are doing PhD, UG and PG and PhD faculties are participated in the poster presentation session. I think it will be yeah, just a minute, I'll share you that. So it will be useful for you. It will be the poster presentation session was there. I think this session you are enjoyed very well, and you have presented the posters actually. So I would like to announce that of a price of the e-poster evaluation. The poster evaluation is done by the Dr. Balasubramaniam T. He is an head professor and head Department of Pharmacology, Al-Shifa College of Pharmacy, Kerala, and Dr. Shanmugaratnam. He is Department of Pharmaceutics, Anna University, Trichy, Tamil Nadu. So this would like to just announce that of your best three, that is first, second, third portions of the poster presentation. The first poster, the first position goes to Dr. Sandeep.
Yeah, thank you, sir. Thank you so
Just a
much.
minute. Yeah, just a minute. Is it visible, right? Is it visible?
No, sir.
Just a minute. Just a minute. Just a minute. Just a minute, all of you, just due to some technical issue, just I'll get back to you now. Few minutes. Very good. Good, that's very good. Very good. Yes. So the first prize goes to that of you. The first portion goes to Sandeep. So he has done the work in that of you. He is from that of in Andhra, Chemburlu Women's Institute of Pharmaceutical Science, Andhra Pradesh. The first portion for the e-poster presentation for the preparation and evaluation of improved state oral disposable tablet during the secretor retired starts in the excipients. So congratulations for you the getting a first position in that of your poster presentation. Congratulations, Sandeep sir.
Thank you, sir. Thank you so much.
Next. The second portion, due to the tough competition, stop sharing. The second portion goes to, just a minute, I'll share. Yeah. So the second portion goes to Haripriya from Nehru College of Pharmacy, Kerala, for securing the second portion in the e-poster presentation, that antenatal use of corticosteroid for the prevent respiratory insufficiency as well as preterm babies. So congratulations, Haripriya, for securing the second portion. And the second portion is shared by Mohammad Afzal VC from College of Pharmaceutical Science, Government Medical College, Trivandrum, Kerala, for securing the second portion. Just sharing the second portion. His presentation is a drug used for that alleviate novel coronavirus infection. Congratulations, Mohammad Afzal.
Thank you, sir.
Yeah. Third portion go to Simrana Fatima of the Sri Ramachandra Institute of Higher Education and Research, Chennai, Tamil Nadu, for securing the third portion in the e-poster presentation. Comparative study of synthetic and natural super disintegrant on the formulation development for the fast dissolving tablet containing macrolide antibiotic by the direct compression method. Congratulations, Simrana Fatima.
Thank you, sir. Thank you, sir.
And the third portion is again shared by Deshna DS, JDT Islam College of Pharmacy, Kerala, for securing the third portion in the e-poster presentation. A review on synthesis of novel heterocyclic derivative of chalcone as a potent pharmacological active molecule. Congratulations, Deshna madam, for your presentation, securing the third portion in that. So by
Thank you,
this, we
sir.
just
Thank
are
you.
going.
Thank
Yeah.
you.
Thank you. And thank you all of you. Congratulations for all that of your 5 participants. That is our first position goes to Sandeep sir. Second question goes to that of your. It is shared by Haripriya and also by Mohammad Afzal. And third position is shared by the Simrana madam as well as Deshna madam. Thank you. Congratulations. Thank you and congratulations for your effort. Thank you. Thank you so much. Yeah. And now I would like to invite to all the participants to give their feedbacks. So just those who you can come voluntarily and give the feedbacks.
Yeah, good afternoon, everyone. This is Sandeep from Guntur, Andhra Pradesh, Chevrolet Hanamaya Institute of Pharmaceutical Sciences. And actually, I want to discuss about 2 topics over here. The first one is the session was very, very nice, and it was a 2-day session. And instead of filling it with me too many of presentations, you have just confined to two sessions in 2 days. So it is a very good session. And second one is the evaluation of the poster presentation. Yesterday it became nearly 3 p.m. But along with Karthikeyan sir and Balasubramaniam sir, Shanmugaratnam sir, they were very patient and they were very nice to all the people. There were no reciprocations or no allegations. Everything went very smooth and very well. And as a pharmacology person, I'm very much satisfied with this conference. And thank you, Sydus Publishing Group and Kalinga Biomedical Research Institute for giving me this opportunity. Hope we will see the same and even more conferences from you in the future, sir. Thank you.
Thank you. Thank you so much, Sandeep sir. Thank you so much for
Thank you,
valuable
Sandeep sir.
suggestions.
Thank you very much.
Thank you, sir.
Yeah. Anybody else they are ready to give that of your feedback? Please come forward and give your feedback. Please. So those who want to give the feedback, please you can come forward. You can unmute yourself and you can give the feedback, please.
Good afternoon. Myself
Yes.
Desna, and I'm very much thankful to you for giving me such an opportunity to present a topic in your that is in your session and webinar, that is in a national conference, and the 2 days program was very well organized, and I was too happy, and I'm still very happy to having this third prize in your conference, that is in the presentation, oral presentation. I'm very much thankful to you, and all the best for your future seminars and conferences. All the very best.
Thank you. Thank you so much, madam, for your suggestion. Thank you. Anybody else, please? Those who
Hello,
want to
sir.
give the, yeah, please, yeah.
Hello, sir.
Yeah, yes, you can proceed. Yeah, please.
My name is Anagha Sudindran. I'm a B.Pharm final student at Ahilya School of Pharmacy, Palakkad, Kerala. First of all, thank you for organizing and given opportunity to attend this webinar. All sections are very enjoyable and understandable. So I hope these sections are very useful or helpful for all participants. Once again, thanks to organizer and all presenters. Thank you all. Thank you so much.
Thank you so much, Anaga madam. Anybody else to give the feedback? Those who want to give the feedback, you can give now, please.
Good afternoon, sir.
Yeah, please. Yeah, please.
Sir, I am Mohammad Afzal from College of Pharmaceutical Science, Medical College, Trivandrum. So it was a nice experience to attend this national level conference on the innovation in pharmaceutical and biological sciences. And especially I am thankful to Dr. Karthikeyan sir for giving me the opportunity to give this platform for my presentation. Also, I'm thankful to all the expert speakers, especially Dr. Balasubramanian sir, Dr. Diba madam, Dr. Karnagara Reddy sir, and Dr. Nirmala Devi madam, and all the organizers behind this program make it successful. Thank you all. Thank you, sir.
Thank you so much, Mohammad Afzal sir. Thank you. Anybody else, please, you can give the feedback. Anybody? Anybody else you want to give the voluntarily, you can unmute yourself and you can give the feedback. Anybody else, please? Those who want to give the feedback, you can give the feedback. So it will help us to improve our things, any deficiency, and we can improve in the future also, please. Yeah. We are conducting this program, the third program, actually in the pandemic period, in the COVID-19 pandemic period. So it was a third program we are organizing, and this was the first conference we are organizing by the Sydus Publishing in collaboration with Kalinga Biomedical Research and Training Establishment, Odisha. So around more than 670 participants were there. So anybody else, those who want to give the feedback? If not, we'll proceed for the next session. Okay, thank you, and we'll proceed for the next. And I would like to invite Dr. Deepa and to give the recap of the 2 days conference. Yeah, please, you can proceed. Yeah. Kumar Kumi, please, you can proceed for the feedback. Yes, please. So, sir, if you want to give the feedback, you can proceed, sir. No problem. Just you can proceed. Kumar, sir, please.
Hello.
Yes, yes, sir. You can proceed. Actually, no problem.
Hello. Hi, Kumar from Karnataka, Srinivas College of Pharmacy.
Yes.
So this is my second conference, which was conducted by you.
Yes.
So such a nice sessions which was conducted by you, and yeah, it's good. Like this lockdown time is more utilized by attending these type of conferences and all. So thank you so much, and good luck for all your future conference and all. Thank you so much.
Thank you so much for your suggestion, sir. Thank you so much.
Hello.
Yes, sir. Yeah.
Yeah, very good afternoon to all.
Yes, sir. Yes, please.
It's a very great experience to listen. This is Dr. K. Srinivas from Warangal. It's the Vagdevi College of Pharmacy.
Yes, sir.
Yeah, I'm working as associate professor here. Sir, very congratulations to Karthikeyan, sir.
Yes, sir. Thank you, sir. Yeah.
Excellent job. So have given some useful lectures to all the students and staff to understand the things is very well. Their lectures are very good and very understandable.
Yes, sir.
That is very nice. Lectures are very nice. You have chosen very good topics. Anyway, congratulations, and Sydus and Kalinga also. Future also we are expecting same. We are doing a lot of efforts to in this pandemic time to give an information to all the students and staff. Very excellent. Congratulations, sir.
Thank you, Stas. Thank you so much for your feedback, sir. Surely.
Yeah.
Thank you so much. Anybody else, please, you can come forward for the feedback. Yes, sir. I think we can proceed for the next one. And I would like to invite Dr. Deepa, madam, to recap the 2 days conference.
Just a minute. A warm good afternoon, one and all gather here. Sydus Publishing India organized its first national online conference on innovation in biological and pharmaceutical science with the theme on recent advancement in pharmaceutical and biological science on 23rd, 30th August 2020. The conference brought together academicians, scientists, researchers, and students of biological as well as pharmaceutical sciences from different states of India, also from abroad, with 670 participants. The conference started off with the prayer. Dr. Deepa MK, coordinator, welcomed the gathering. Dr. Karthikeyan M, convener and editor, Sydus Publishing India, inaugurated the event, followed by the inaugural speech. The first scientific session, the topic, polycystic ovarian syndrome, risk factor for type 2 diabetes, was delivered by Dr. T. Balasubramaniam, Professor, Head, Department of Pharmacology, Al-Shifa College of Pharmacy, Perindalmanna, Kerala. The second session, the development of nanoparticulate drug delivery formulation and its impact in disease management, delivered by Dr. Deepa MK, followed by scientific session. Poster presentation of various participants were held. Professor Dr. T. Balasubramanian and Dr. A. Shanmugarathina, Associate Professor, Anna University, Trichy, Tamil Nadu, were evaluated the e-posters. The second day started with the third session by Dr. Karna Reddy from Pharma Industry. His topic was Scope of Regulatory Affairs. In the first session, the topic Advanced Technique in Bioscience Research was delivered by Dr. R. Nirmala Devi, Assistant Professor, Department of Biochemistry, Avinash Linga Institute of Home Science and Higher Education for Students, Coimbatore, Tamil Nadu. This conference scientific session has given some idea to spark for the young research to direct the research in advanced biomedical and pharmaceutical innovation. All the scientific session closed by 1:30 p.m. followed by validative function was arranged. Dr. Subhendu Mishra, Director, Kalinga Research Institution, was given the guest speech, followed by Dr. Karthikeyan M, convener, declared the Best Poster Awards. Thank you for your patient listening. Now come to the last session, that is vote of thanks. I would like to invite Dr. Karthikeyan, convener, for the same. So please.
A warm good afternoon, all of you. It's my immense pleasure to propose a vote of thanks in the occasion of valedictory function of the national level online conference. It's an honor and privilege for me to express a gratitude to Subhendu Shekhar Mishra, the co-coordinator and the director, Kalinga Biomedical Research Institute and Training Establishment, Odisha, for the endless support and constant encouragement throughout the event. It's my privilege and a great joy for me to extend the word of thanks and respected speaker, Dr. Balasubramanian, Professor and Head, Department of Pharmacology, Al-Shifa College of Pharmacy, Perindalmanna, Kerala, for his valuable guidance and support and accepting this invitation as a speaker as well as a poster evaluation in the current conference. I take this opportunity to express a heartfelt thanks to the respected speaker, Dr. Deepa M.K., Ashokramani College of Pharmacy, Kolhapur, Maharashtra, for sharing the research experience. Thank you, madam. It's my privilege to express my gratitude, respected speaker, Karna Reddy from the pharma industry, for taking the time off and the hectic schedule to being today with us for sharing the experience and the opportunities for the regulatory affairs for the BPharm, UG, PG and MPharm students. Thank you, sir. Extend a thanks to the respected speaker, Dr. Nirmala Devi, madam, Assistant Professor, Department of Biotechnology, Avinash Lingam, Institute of Home Science and Higher Education for Oman, Coimbatore, Tamil Nadu, for a kind cooperation, accepting an invitation and deliverable the wonderful session and useful techniques for the UG, PG and PhD, madam. Thank you, madam. I would like to express my deep gratitude to the Shanmuratnam, Associate Professor, Anna University, Trichy, for the valuable presence and being a post evaluator for the e-poster session. Thank you, sir. I also acknowledge that a contribution all those who have presented e-poster and the national level online conference. Thank you all those who have contributed the e-poster and the presented the paper in the e-poster session. Thank you for all the e-poster presentees. On behalf of the Sydus Publishing and myself, I would take all this opportunity to thank all the participants to being with us for the last 2 days, with us for the morning 9:30 to 2 o'clock. And yesterday we have extended time up to 3:30. Even the time was extended. All the people are with us for the last 2 days and today also. So I'm thankful to all the participants to being with us for the last 2 days and their patients also. I'm very thankful for, and also the participants from all over the India as well as abroad for their enthusiastic and active participation. Thank you all the participants. In the closing, I declare that the online conference is end now. Thank you and all. And I would like to request all the participants to rise here or the national anthem for the ending of the ceremony. Thank you and all. So just for 1 minute, a 2-minute session, I would like to ask all the participants to rise for the national anthem. much for all the participants.